Good morning, everyone. Good morning to all those who are online. It's really nice to be together here on this warmer but rainy day at the Arboretum. And I wanted to introduce myself. My name is Melissa Poe. My pronouns are she, her. I'm the Assistant Director for Outreach at Washington. I know many of you were here yesterday, and this is day 2 of your workshop. So welcome. I wanted to open today's meeting with an acknowledgement to honor place, and I've prepared a couple of words. I'm going to share those with you here. Let's start this morning by acknowledging that we are standing on the lands of the Coast Salish peoples, whose ancestors have resided and have stewarded the lands and waters since time immemorial. The University of Washington, which Washington Sea Grant is part of, including the Arboretum, acknowledges the Coast Salish peoples and whose homelands touch the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. And we give our sincere gratitude to be here in this really beautiful place. I hope at lunch or already this morning you've had a chance to walk some of the grounds, appreciate the plants that are here and the bird and other kinds of life that's supported by this important habitat. So SoundToxins is a network with monitors, yourselves, and stewards who are distributed throughout the Salish Sea. And it's also important to acknowledge the work you do that honors place. And we give our sincere gratitude to you all and for the work you do at Toxins programs. So SoundToxins is part of Washington Sea Grant, and it's a partnership. It's a partnership with all of you and with other agencies. This vision of doing community science networks and monitoring plays an important part of the Washington Sea Grant program, and it helps us achieve our mission. So I wanted to tell you a little bit about the Sea Grant mission, which is to help people and marine life thrive by supporting research, technical expertise, and educational activities that foster the responsible use and conservation of ocean and coastal resources. This SoundToxins research and monitoring partnership is really core to our outreach program. And in particular, SoundToxins contributes directly to one of our goals that the ocean and coastal resources are managed applying sound science and local and traditional knowledge through ecosystem-based approaches. So by training and supporting monitors throughout the state, and by providing information about environmental conditions and other phytoplankton to managers, we are able to help support other goals that are key to Washington Sea Grant's mission, including that coastal water resources sustain healthy human and coastal ecosystems, and also that we are protecting safe seafood and accessible seafood. So it is this strong partnership that you all embody that is at the foundation of SoundToxins, and you provide this crucial information to monitor HABs and to minimize the health risks and economic impacts that are associated with our beloved water and shellfish resources. So thank you all for being here today and to the SoundToxins program. This is an exciting annual meeting and an important time for us to get together, secure the work we do, and to hear what lessons have been learned. So today's program is going to have a lot of great information. But I wanted to give a special thanks to Michelle Lepori-Bui, who manages the SoundToxin program. And Michelle, I have a little something for you. So come up and receive this gift and receive our appreciation for everything you do to steward this network. Awesome. It's beautiful. And Biotoxins. Thank you.
Thanks, Melissa. Okay, what's next? Bowser Sea Grant, welcome. We talked about this yesterday for those of you who were able to join us, but for anyone who's new here today, we wanted to share our Washington Sea Grant Code of Conduct, which will apply to our meeting today, but also in all the work you're doing through SoundToxins as associated with us. So please treat everyone with respect and dignity, share perspectives and offer feedback openly and honestly, and be open to experiencing new concepts and differing viewpoints. Work to ensure that others have opportunities to speak and share. Take responsibility for your actions and impacts, acknowledge harm when it has occurred, and apologize. Report perceived unacceptable behaviors directed at yourself or others using reporting options. You can contact us, or also you can see the full code of conduct by scanning the QR code here. I think it's also available on our website if you need those resources. So the next bit of time that we have is for our station introductions. So yesterday we just went around the room and introduced ourselves and shared very briefly how many years of experience we had. But this year, if you want to, we have like a little bit more time, so you, not too long, but tell a few minutes about like kind of your site and kind of, you know, what for some of you might be part of the program, but do kind of other things in addition to it. And some of you might have, you know, if you're new, you can just say hi, but if you've been here for longer, talk a little bit about your background or experience. And this part is meant to kind of connect the network. You know, a lot of you have kind of connected already from from different sites to kind of share resources and connections. So this is kind of an opportunity to like chat a little bit more about that. Still try to keep it not too long, yeah, so that's our time to do that. So I guess we'll start with the folks in the room. I'm not sure how well that worked yesterday for people online to hear what people were saying, but we'll try to project our microphones just at the podium, not like walking around. And then if folks online want to unmute themselves afterwards and introduce themselves, we'll have a bit of time for that. You can also share in the chat and I can read it out if you want. But we'll start in the room. Would you like to start over there?
Sure. I'm Julie Wage from Camino Island, and Camino Island State Park is where we do our this time of year it's without a dock, so we have to wade in. It's interesting, and so far not too stormy. And then I also have a mussel cage that I monitor, use for monitoring.
Nancy?
Nancy Stephens, Port Angeles. We got the program just started. This coming July, it'll be 3 years. I was told by Michelle earlier when we got trained that Port Angeles had been one of the first stations, but then that lapsed. And so we got that going again. And I'm also on the Clallam County MRC, so I'm kind of the MRC representative volunteer for the SoundToxins program. And it's run through the Faro Marine Life Center in Port Angeles.
Do you want to just go down the rows next? So maybe Alicia can go next and we can go that way and then we'll jump to the side.
Hi, I'm Zach Lee. I work at Hotel Shellfish. My station is Curly Lagoon. And I've been doing it for 40 years now. It's very challenging, yeah.
Hi, I'm Leslie Hutchinson, and from Orcas Island, East Sound. We have the county dock as our site. We don't have our float at the moment either, but we don't have to wade in. We can drop it from the pier. So that's we've been doing that for several years. And you can see this from the books.
And we also used to have a station at Long Lake Kings just down the street. And unfortunately that's gone. So the people that were doing it quit, and I guess it never got restarted.
Yeah, I'm not sure about that. I think was like right around the time I just started, but always,
was always felt that they weren't that far away from us, so.
yeah, I mean, I think since you guys have a site there, it's maybe not on the top of our priority list, but also if you're ever in contact with those folks and they want to start again, we're definitely available to chat.
We did that, find more heterostigma, but besides that, we were pretty much.
Yeah. Yeah, those two sites were fairly close together, I think, but
Other than,
yeah.
oh, Lisa.
Thanks.
Um,
Jackie.
And we sample 3 different types, we sample brown and the Dyson. The other team member is my husband is actually online. I see him right here. So, and I'm here and not, and,
Good morning, my name is Mary Kelly. I am, my site is Quartermaster Harvard on Vashon Island. It's my first year doing it by myself. I worked for quite a few years with Carissa, and she's retired. So I've learned a lot. I have a lot to learn. I'm looking forward to seeing the seasonal stuff, the changes, and like that.
Jackie Fuller. And go on Tuesday mornings, and our team is involved in not each of us specifically, but among us all, we're also in the Greengrove, monitoring, C-star monitoring, the muscle cages, and raw. I think all but one of us are in beach natural, so we had it here.
Awesome. Yeah, you're next.
Our private with over we monitor Semion Marina and Birch Bay Marina, which is like right on the Canadian border. We're on that last site until you can see White Rock, Canada, right from our monitoring site. And something kind of interesting this past year, our recreational shellfish harvest closed pretty much May through September for Alexandria. So, yeah. And our samples last year. I'm Lee Beagle, volunteer with SoundToxins through the Fort Townsend Marine Science Center in Fort Townsend, and also volunteer for the Department of Health.
Brad Beagle, also in Fort Townsend, usually Discovery Bay, and I just help carry them net to the water. Get them bottled in water.
Why don't we come to the front here? You want to go, Tracie? And then we'll get the people standing in the back.
Morning, everybody. Very morning, again, like Department of Health. Just so excited to be here. So grateful for all of your work.
I'm Amy. I work at the Shellfish Institute and clinical and veterinary. Always exciting.
It's Carlson, and with the Fort Townsend Marine Science Center, I coordinate the Cyst there, and then I'm also with the Jefferson County Marine Resources.
My name is Abby Anderson. I'm involved with the Tomato Group, and then in the beginning, I moved right after that. So I still participate some online when they have online stuff, and then it's refresher every year. And I use this information. It's super helpful in the work I do with residential.
John Vitale, I'm another Fort Townsend Marine Science Lab person. I sample at Discovery Bay and Marshall Island near Fort Townsend. I'm also with the Dungeness Crab Monitoring Group, which is fun in the summer, and lately I've been looking on a clam project, I forget the name of it, but we measured clams trying to look at, how bad that, see, the acidity is affecting the shell.
Doug, you wanna go?
Oh, sorry. Doug Johnson, I'm the newbie in the bunch, came out of an engineering background. I was pre-med before and want to get back to something that's more fun, more realistic, more water time. So we're in the Hood Canal on the west leg, so we've got a cap. We run up and down the coast, and I'm hoping that if they don't throw me under the bus, that I can hook up with the Port Townsend guys.
I'm Emily Bjornsgaard at the Mass Center, so we stand Poverty Bay right off of our dock, which is great. Well, it's king tide and we've got water on the dock. And I've been with SoundToxins about 2 years, but I've been doing phytoplankton things since about 2018. And at the Mass Center, I'm the lead aquarist, so I take care of all the animals. So.
Hi, again, Melissa Poe at Washington Secret, and I'm here supporting the Michelle and Melissa and the SoundToxins programs.
I'm Kira. I work at the Sailor Sheet Research Center up in Bellingham. We sample Fairhaven Dock, Bellingham Bay buoy if it's not too choppy, and then we spray a point if the dock is there. Currently, we don't know where it is.
We covered, and I'm visiting from Florida. I'm part of the Florida Fish and Wildlife Conservation.
Yes. Go to the front and back.
Caliber, sitting back in the Strait Malays. I am in the Port Angeles team with Nancy up in the front row. The Faro Marine Life Center. I'm staff there, kind of an aquarist in training, and also dealing with our citizen science programs. Also staff for the Palm County Marine Resources Committee.
I'm Rio Foster with the Fleet Tribe, and I've been a specialist for about 12 years now. I sample the Bush Beaches, Laylock, and Reed Beach.
Jeff Adams, I'm another Washington Sea Grant colleague here supporting Melissa and Michelle. Moving around miles today before I go cut bait for crab team, the science program with Washington Sea Grant. So it's wonderful to see you all here and see all your faces.
Hello, Anthony Odell, University of Washington, ORHAB out of Forks. Work monitoring the outer coast, and now I'm trying to integrate a little more with Michelle, work with SoundToxin, but with ORHAB, I've been doing that for about 25 years.
Everyone, my name is Melissa Petrich. I'm pretty new. I'm a Washington Sea Grant and the water quality specialist for South Sound and Canal. And I'm helping Michelle with Cyanotoxins, so I'm excited to be here.
Okay, thanks. And then folks online, if you would like to unmute yourselves and introduce yourselves, now is a time that you can do that. Not mandatory, but if you'd like to. Okay, I see Rick's camera on. You want to go ahead, Rick?
Sure, I'll go ahead if you can hear me okay.
Wait, hang on, let me turn up the volume here.
Hello,
Okay.
hello. Testing 1, 2, 3.
We're good in the, okay, go ahead.
All right, Rick Beauregard. I've been stalking Alexandrium catanella since about 1970, formerly known as Goniolax tamarensis, back in the Gulf of Maine. And after a 50-year hiatus, we started Drayton Harbor HAB Hunters, which is now part of the Whatcom County MRC projects. And so we've been doing it for the last several years up in Drayton Harbor, the tippy top of Puget Sound within the United States. And glad to be here. Sorry I couldn't be in person.
Problem, glad to have you online. Anyone else online would like to? Let's see, I got Kathy, who is part of the Camino Island monitoring team, and Chris, who was mentioned as part of Kitsap by Jackie. So welcome. And we have a few folks joining us today too that are going to be presenting, including Avery Tatters, who is from US EPA. And I see a couple other of our presenters who will maybe introduce themselves when they present later today. So we can pause on the introductions unless anybody else wants to jump in at this time. Okay, great. Well, we're a bit ahead of schedule, but I think we can just go on. That way we'll have more time for questions and things. Tracie, are you ready to go? Okay, awesome. Let me switch the slides really quickly, and then we will have Tracie with her presentation. I could probably use a couple extra minutes, so that'll work then. Okay. Doing some logistics here. Start the slideshow and then share screen. Yes, we can make that happen. Hold on. Hang on, let me see if it'll work. Yeah. Oh, yes. As a quick question. So we did our supply exchange this morning. Just wanted to double check if everyone was able to drop off their vials in the back of the room. Because Jeff is going to take them away now. So
Should I do it right now?
no, pick up, we can get to you at like lunch or the break. Just taking away. And then if anyone had formalin bottles that needed to be refilled, you've all dropped those off. Okay, and you can pick those up later too, just the vials that are gonna. Okay, awesome. Thanks so much, Jeff. And then just on that note, there is extra things that you can pick up if you need them in addition to the vials. So we have pipettes, like little vials of Windex and Q-tips if you want them to kind of keep your microscope clean. And also I forgot to move the Chem wipes from the back to the front, but if you want to get a box of Chem wipes, there's a bunch in the back. So feel free to grab those, and there's cover slips as well. And if there's anything else you need, reach out to us and let us know. Okay. So you can have this here, but then I think to advance the slide, it might be a bit weird. I can live without him. Okay. Probably. Okay, well, hang on. Let's see if we can, let me see what I can do. Oops. Okay, I'm sorry. Okay. Well then, here you go.
Okay, good morning. I'm Tracie again. Thanks for giving me some time and kind of letting me start the day talking about our problems, because we're going to talk about a whole bunch of other harmful algae problems as the day goes on. So our friends, I won't go into it too much. If you're unfamiliar, the syndromes sound like their names, which is really useful. The one-time science made it really easy. We've been monitoring for Alexandrium and PSP since 1957, diuretic shellfish poisoning since 2012, and pseudonychia and domoic acid since 1991. So we have a pretty good history with these fellows and whatever pronouns they'd use. It's hard to not anthropomorphize them when you see them alive on a slide, right? One thing that you might not be familiar with is the Shellfish Sanitation Model Ordinance. And so for Washington and any other state to participate in shellfish trade, we have to meet these requirements. So there's a lot that goes into it, but we have to have administrative authority over all aspects of shellfish growing, risk assessment and management. So that's things like illnesses. So norovirus is a big thing in the news right now, but biotoxins in particular are important. Dealer certification, growing area classification, and biotoxins falls under that. And then we work with the FDA to be evaluated so that all states are acting in congruence, and then also for international trade as well. So our management program, we have a contingency for HABs that are toxic but aren't known locally. And again, that's where Kate came in to talk to us about Karenia. And then our very own toxin former, we have our own toxin in Washington, AZA 59. It's because they're teeny tiny. So these guys max are 8 microns tall. So they're going to go right through your net. They're kind of a pain, but if there's enough of them in your sample, you're going to notice them. And then we have our management plan for our HABs with lots of history. So our management plan is based off of our Sentinel Mussel program, which many of you are a part of, which is really critical. From there, we can actually expand testing to weekly from our normal biweekly schedule move that into expanded commercial monitoring. So we get that trigger from our mussels. All the commercial farms are now sending in samples, but we can really distill that down based off of that Sentinel program. We have specific plans for gooey duck and my personal favorite, razor clams. And then once we start to see toxins in commercial species, we can actually go to lot testing. So every individual harvest is tested for biotoxins. All of this is supported by your work, by that phytoplankton monitoring piece. It fills in the gaps between days, between weeks. It lets us know when something's going crazy. And it was incredibly important for us being able to reestablish trade with the European Union. Washington is able to export shellfish because of phytoplankton monitoring, which is the work that you're doing. So we test quite a few samples. It varies each year by what toxin we're seeing. So if we have a bloom on the outer coast, we're going to see a ton of razor clams, probably a bunch of Dungeness crab. If we see really bad toxins in the Central Sound, we're going to have a lot more gooey duck. But really the backbone of everything is our mussels. So 61% of our samples last year, 13% were oysters, which means there was a lot of commercial sampling. And then 10% for gooey duck is pretty standard because they're normally harvested subtidally. And so what's happening at the surface in our mussels and our phytoplankton doesn't really match. So most gooey duck is tested on a weekly rotating basis. Now, the work is really spread out too. You know, Aaron and I are running around. We're part of that state agency chunk, which is about 17%. DNRs included, Fish and Wildlife. Tribes are 23%, collecting samples to inform their own harvests. Volunteers are 14%, local health is 27%, and 19% of all that testing that's done is really that commercial sampling piece that's informed by the phytoplankton and the sentinel mussels. So just kind of backing into how our program works a little bit, and this is just a great example. So Squim Bay is one of those crazy hotspots, much like Disco or Bellingham Bay. And of course, if you know Neil Harrington, he is just extraordinary at phytoplankton monitoring. So it's a great story. We also have a lot more sampling than you'd usually see during this event because Squim Bay had domoic acid and diuretic shellfish poisoning at that time. So there's a lot of data gaps that are filled in because we were on top of everything. Now, this year in particular, things got screaming. So almost 50,000 cells per liter. And you're going to see your cells per liter on your right-hand side, and then the PSP toxins in the shellfish is going to be on the left-hand side. So we're going to just kind of isolate down and focus on what's happening during this event portion. So this is the whole story. So I've got the different shellfish as different colors. We've got our little tiny red balls as our Alexandrium. I did try to make a tiny Alexandrium for that one. And then our action level is 80 micrograms, and that's in shellfish tissue. So we see kind of end of May, which is kind of our typical PSP period. There's no cells, super great. But we see a little uptick somewhere around mid-June. So that starts the weekly mussel sampling, right? Those things work together. And then things kind of stay stable. They're jumping around. There's cells in the water, somewhere around 250 cells per liter. Not crazy, but that can change at any time, as we all know. And then the mussel toxin concentration dips down again, but we're still on weekly sampling because there's still cells present. As we move into September, that's a lot of time of sampling on a weekly basis. We see the mussels, even though the cell counts are low, pop over that action level. So now we put in a recreational all species closure. And again, this is paralytic shellfish poisoning, which matters later. And then the weekly commercial testing starts. Shortly after, we see the cell counts drop down again, and then they just go flying. Remember, we're at 50,000 cells per liter from somewhere around 200. And that happened overnight. And then we see that our commercial species go toxic as well. So because that phytoplankton monitoring was happening, everybody knew, and the weekly mussels, everybody knew that they needed to hold their product and make sure that they waited for a test result. So we would have been on weekly testing at least, maybe lot testing, but every commercial farm was making sure that their samples were getting tested before they put them on the market. So no recalls, nobody got sick. It's an amazing testament to how this program works. But we see this continue on. 350 micrograms per 100 grams tissue for manila clams and oysters is actually pretty screaming. Sandwich Bay is another spot where we see 1,000 micrograms and oysters, not that infrequently anymore. But things finally start to quiet down, kind of end of October. Cell counts dwindle, but we're still seeing a lot of variation in the mussel toxicity. you know, you'll see it in your phytoplankton samples. There's a lot less food out there. The shellfish are kind of going dormant, so they'll hold on to those toxins. But it still takes a while till we can upgrade the commercial shellfish, right? And so that finally they can start harvesting again about November 1st. And we see a lot of variation again in our mussels, but we have a lot of confidence that the cell counts are down. Again, this is a little bit wonky because there's diuretic shellfish poisoning at this time too. So there's another closure happening, but it isn't until, middle of December of that year that we were able to do the rec all species upgrade. And then a butter and varnish clam closure stays in place. We know that those species hold on to toxins for very long periods of time, up to years, as the Camino Island folks can tell you. And so it does take a long time. So we'll probably test those maybe 5 months later. We can do some kind of fun deprivation calculations, roughly estimate when we should put effort into collecting those samples, because butter clams are hard to get. Varnish clams, not so much, but butter clams take a lot of effort. So that's kind of our piece of the puzzle. With that rec upgrade, the second we have information from a sample, this is where we put that information, is the shellfish safety map. So if you haven't seen it, please check it out, share it with your friends and neighbors. I'm from the coast, and so I didn't know about this till we started working here. We always just listened to the man Dan Ayers. If you're a razor clam digger, you'll know that name. But we actually put all that information in real time on this map: pollution closures, Vibrio closures, but mainly it's used for biotoxins. It's a really great partnership with Fish and Wildlife too. So if you click on any of those beaches, it'll tell you what the harvest season is. Usually it'll give you a bunch of information about where to dig and what to dig. It's, I'm just amazed all the time, and I had nothing to do with it. I just changed the closures in my living room while my dog's barking. So now we'll dig into a little bit about Washington. So as all of you know, there's a season to Alexandrium, and we very much see that in our samples. And so really for Alexandrium, and this is the number of samples above the action level, not necessarily the toxin level, but it kind of shows you that seasonality. And I've taken butter and varnish clams out of it, and any coastal samples, because razor clams hold on to domoic acid, butter and varnish clams hold on to PSP. So you really see things start to pick up in May, but there's a distinct kind of peak July through October. For diuretic shellfish poisoning, except Bud Inlet, where Amy's at, really we see a peak in July and August, and then another one in October. For domoic acid, if you're going to see it produced by Pseudonychia, the little bit of information that we have for Puget Sound tells us that that's really going to happen in September and October. I would say we see a little accumulation for some reason in the Whidbey Basin kind of in the spring. And I think it was 2 years ago we had our last little blip, and it got real scary. And for some reason, manila clams tend to pick that up quickly too. So if you've got manila clams on your beaches, you never know when you'll get a call. Little frantic. So looking at our hotspots. So we only have 12 years of DSP data, new kid on the block there, but we have some really distinct hotspots. Bellingham's Bay is one of them, Discovery Bay, Squim Bay, a little bit of the North Coast, and then Bud Inlet, just king of DSP. Woo! Domoic acid, a little bit in Squim Bay. There's been 2 events. We had one in Penn Cove and Saratoga Passage or Holmes Harbor, and then another one in Killisett Harbor. But really, it's predominantly a coastal issue, which is seen kind of throughout the world. Even if cells are there, it's really that outer coast, that true marine influence where you see that toxin production. And Kate's looking at me like I'm misinformed, but she's also my hero, you guys. So, and then for PSP, it's been just about everybody, everywhere. And I didn't explain these to you, and I'm so sorry, but the darker the gradient, the more years there have been closures, not necessarily events, because you can't always tease that apart. So we're just saying years with closures. But Bellingham Bay, Discovery Bay, Squim Bay have had closures almost every year, if not every year, since we started monitoring in 1957, which is insane. The Central Basin has been known to be a hotspot. It's kind of quieted down the last few years. And now we're seeing it pick back up again. So Lord only knows. And then Hood Canal has mostly been spared, a lot of the southern Puget Sound, but not always. So just kind of stacking things up, 2024 was really quiet for domoic acid, which was fantastic. There were no closures for the first time since 2014. Really good news for, again, the razor clam diggers on the coast. We have had a little bit of blooms within Puget Sound and closures for domoic acid, but we seem to be turning it into a quieter period, and we're hoping that that lingers for us, like the mid 2000s, early 10s, and we get a little bit of a reprieve out there. For DSP, last year was surprisingly quiet, except for the South Sound. We didn't have any commercial closures. We did have 4 recreational closures, but we did have quite an unusual event that started, would have been about November of 23. And then in January of 24, it just went screaming. It spread to Pickering Passage for the first time, Eld Inlet into Henderson, which it only had done once before. Really caused a lot of disruption for a lot of farms that had never experienced it before. And I have a feeling that's why there's a few more faces in this room, is to help monitor phytoplankton in the South Sound. Bud Inlet, so right the epicenter of that zone, still holds the national record for the highest level of diuretic shellfish poisoning toxins at 250 micrograms, and that was in 2016. And then the same bloom was the highest level in 23 and 24. So last year our highest was 129 micrograms. We saw another similar event just recently that we're still working through with Bud Inlet, and then the bloom seems to kind of spread out from there and then impact Totten and Eld and Henderson. So really productive shellfish growing areas. PSP, kind of see it everywhere. Strait's a hot spot. We've had a couple quiet years. Last year, I would say, was definitely not. The outer coast was an issue, which was pretty unusual and got a lot of press. The Strait, through the Whidbey Basin, North Basin stayed a little bit quieter, which was a huge relief. But still was problematic per usual. But we did have 20 commercial closures and 26 recreational closures. And that's sort of events. So how many times we made phone calls, not necessarily how many water bodies were impacted. But in the grand scheme of things, it really didn't stack up very high for maximum toxins. It was 1,513 micrograms in mussels from Kingston. So Kitsap crew, you were probably having eyes on a lot of Alexandrium. But in the grand scheme of things, it just really wasn't that toxic. But we saw over 1,000 micrograms, which is considered lethal, in a whole bunch of areas. It was pretty widespread that those blooms were pretty intense and intense enough to make people severely ill or die. We did have one interesting result that I didn't put it up here because it doesn't match well, but it was scallop viscera from the Strait was 5,434 micrograms. It's really commonly thought throughout the world that if you just eat the adductor, it's safe. The testing that we did with samples from the Washington Freedivers Club, so these are people just holding their breath, diving down and collecting these. They like to eat them, but the viscera from every sample that they sent in was toxic as well. A little bit about the coast. I don't want to go too crazy, but this was a really extraordinary bloom. And who heard about this? A lot of people. Okay. It really kind of came out of nowhere. So April, we had no toxin detected. It was really nice and quiet. And then just about the time we headed into Memorial Weekend, things picked up. So we collected some mussels off the jetty. They were at 383 micrograms, where they were about 41 the week before. Lincoln City crept over the action level, so in Oregon at 148 micrograms. We were able to close Grays Harbor recreationally and started a commercial notification where we did get commercial samples in. They were below the action level, thank goodness. But over that weekend, right, we have a 3-day weekend, everybody's excited and on the beach, there's really great low tides. I'm looking at my email because I have a biotoxin alert, my personal phone, and Oregon's got all these people getting sick. They're closing beaches, they're doing the thing. I'm literally sampling on that jetty, begging people to not eat the mussels. We have a mussel closure on the coast all year round. Health says it's never safe because it's so hard to monitor. Fish and Wildlife has a seasonal closure. And so it's April 1st through October 31st. So people were still harvesting. We had signs everywhere. It was a really big thing. Nevertheless, people were harvesting. So Oregon got hit really, really hard. We went into emergency sampling mode. Fish and Wildlife was out there over the holiday weekend sampling recreational beaches, drove them to the lab so they were ready to be tested on Tuesday. Those samples were fine. We get the commercial samples in, they're not. So it caused a really big recall, a really big shutdown, a lot of bad press, really came out of nowhere. The highest levels were definitely found in Central Oregon, so I kind of tried to plot everything here. And there's more data through the weeks that go up, but this is really the peak of that bloom. And Oregon, so Seal Rock, not very far as the whale swims, like 51 miles from our border, was at 5,500 micrograms, and that was in meal remnants of some people that got sick in Oregon. It was really unusual. The bloom developed really rapidly. It seems to have appeared offshore and then come into the bays. There were 40+ people that got sick in Oregon, and 7 of them were hospitalized. I mean, that's on a ventilator. It's paralytic shellfish poisoning. That's how you go. No illnesses were reported from Washington shellfish. I keep thinking about this couple I talked to on the jetty that morning on Sunday, and I'm like, maybe I saved them, and let's hope so, But there were 2 people from Washington that went to Oregon and harvested mussels and did get sick. So we've actually been working with Oregon as well as BC to make sure that we're sharing each other's maps. Maybe people have more information in the same place so that they can get it when they need it. We learned a lot. Lab capacity was really tough at that point for testing the shellfish themselves. So we've been really working hard on a new assay that it should be able to do more samples that will be more adaptive. Vera and the ORHAB group have been working really hard on different screening kits. We've been working to reestablish our connection with poison control, which was really big in preventing more illnesses in Oregon, was that people had each other's personal cell phone numbers and you can really adapt. So we've been meeting with poison control to try to get ahead of that as well. And unusual uptake dynamics. One of the things that really complicated this was our sentinel mussels were less toxic than the oysters. And really, we typically see a progression from mussels to other species, but something about this was different. Next steps, really cool. One of my heroes, another one of my heroes, Dr. Cheryl Greengrove and her teaching partner, Julie Masura, have come out to my neck of the woods to do cyst mapping for the very first time in the coastal bays. And we've been working with industry to update our management plans. So a lot's come from this. Going back to the shellfish sanitation program. So again, neurotoxic shellfish poisoning, NSP caused by Karenia brevis and others, is on our contingency plan. So we have the same requirements of our management plan, but for HABs that we don't usually see. Again, that's phytoplankton monitoring is our early warning. And then our plan previously was to send shellfish to the FDA if we had an issue. Well, we had an issue. So in late August, we had a sample come in from the San Juans where the mice were behaving oddly. And it was reported as a mild PFP case, but I didn't know it was mild when she was calling me, telling she was experiencing symptoms. She was okay. She called me when they discharged her from the hospital, and it was huge relief when she called back. And then right around that same time out on the coast, there was a Fish and Wildlife employee that reported kind of getting the headache and migraine from maybe aerosol exposure, something that they said they never experienced, and they work on the beach a lot. So it was really interesting. And then Micah Rogers with Fish and Wildlife, who's extraordinary if you haven't met her, and Anthony are reporting these super weird cells that we're calling CF gymnodinoid something, right? We're like, all right, that's a lot of the pieces. So we call Kate, and Kate's helping, and Noah's helping. And really, Kate was our savior through all of this. And we were able to get the shellfish samples that were suspect or from those areas off to Kate and her team, and they ran an ELISA. All those samples came back with no toxin detected. And we've been trying to ship samples, the water samples that Micah and Anthony collected for us that were preserved with lugols, not formalin, to Kate's lab to actually do some genetic analysis. So to be determined. We still don't know what we got, but we got something weird. And then of course the focus training here, and it is so amazing to have Kate here with the cultures. It is just such an honor and a privilege to have something like that. They're weird little boogers. And even to have her share her knowledge about how hard they are to ID, how much they change shape, especially in an environment like ours where they're probably not happy. And that kind of leads me to what it is to know a place, right? And so you know a place like most people will never ever know it. And for someone like Anthony, when you see a little weird ball that might be Corenia, or for Micah, or for even me, for any of you, you know what you haven't seen before. And so if you show up to your sampling site and it looks like this, you're like, wow, that's weird. And then you take your net toe and you're like, whew, that's something. And then you get in your scope and you're like, well, that's a dense bloom. You know, it's something like 3 million cells per liter. But you know what it is. It's not going to kill you. It's okay. We're moving on, but it's interesting. And then the internet starts going crazy, right? And the clams are stained. And I was just out there digging that night and I'm like, I know what this is, but my sink's bright red. It looked like molasses or barbecue sauce. But you know, the internet's in kerfuffle. We just had a paralytic shellfish poison event, like red is red, right? And then industry starts calling and something's going on too, and they can't sell their product and they don't know what it is, and everybody's alarmed, but you know what it is. And so when Michelle's calling, or not Terry, I'm sorry, bye Terry, I miss you. Jerry or myself, we're calling and we're worried and we're connecting those dots, and industry's coming to you. You know what that is, and you know that place, regardless of if you're seeing something like this or you're seeing nothing, which can be a little boring, but you get to spend a lot of time identifying what you have. All of that means something. So even if it is one of your priority species, something like this tells you so much about the environment and might just save a lot of people a lot of stress and a lot of headache, and really help support an industry and navigate what is in so many places the lifeblood of our communities. And I just really want to thank you for your time and your space, and for all of your alerts or lack of alerts, for filling in your little dots, because it tells us where we can go, what we're missing, what we can prioritize. And really, it's the backbone of all of this and what gives us space to make really sound decisions and make sure that people aren't getting sick, and also just able to eat shellfish in a good way. Thank you. I don't know if I have time. Anybody? Yes, please.
So when you were showing the commercial fishery counts of what was being reported, did you say those were just the call? Those weren't the number of fisheries impacted? Or were some?
Yeah, okay, that's,
more than once?
yes, exactly. So that's just the years with events, and then the shape files that I use there are sort of water body shape files. So it doesn't represent at all the number of fisheries impacted, because this growing or shellfish production is so different throughout much of the Puget Sound. And there are a lot of areas like the I-5 corridor where there might not be any, but we still monitor because those help inform that space, that water body. But yeah, if you had to put we actually don't track how many phone calls we make, but like I could pull how many closures we had in the area and tell you how many people were on the list that we called. But it gets pretty darn exciting. About 4 p.m. when we're getting those PSP results, nobody wants to answer the phone. They're all heading home for the day. And we're like, we need to talk to you. So, I mean, just last week, I was running around Westport at 8 p.m. on the cell phone trying to get samples from people, and they got lost in the snow by UPS. So I drove them this morning. We got it. Yes, please.
When you talked about the mouse that got sick in San Juan, was that just like a random mouse running around, or was that a test mouse?
That was a test mouse. So the gold standard for PSP is still the mouse bioassay, but we're working really hard to move away from that. The lab is working really hard. Again, the ORHAB group, it's really an amazing partnership, but it takes years to be able to validate something like this. And then for our specific species of shellfish, we have our own razor clams, and then the gooey duck that are so important, even Pacific oysters, and butter clams. So it will be a bit, but we're all very excited that we're moving that way.
Is that
No, we have the public health lab right up the road here in Shoreline.
you just said
Oh, sorry. was, and no, it was a small outer bay. I have been hoping to connect with Friday. It was not San Juan Island. I think it was Orcas on the northern side. So, oh, they shipped the samples to us.
Right.
San Juan County Health Department.
Oh.
Yes, it would actually be really nice to connect you guys, or at least you. Yeah, let's work that out. I think it'd be really valuable.
They must have some kind
I-
of. It's on 10-1.
Yeah, so our partner at San Juan County Health actually every other week takes a ferry to each island, grabs all of them, and then couriers them.
So somebody's.
Mhm.
We don't even know about that. Okay, well, there's something to work. on.
Yeah, it's not really even a network, it's just William.
It's William.
It's just William. Yeah. Well, yeah, let's connect you, and then also with Amy, who sends in the mussels from East Sound. Yeah, let's connect. That'd be amazing. I'm sure she'd love more partners. It's another reason I love this meeting so much, is we get to connect the dots there.
Thanks for asking that.
Yeah, thank you. You know, it all is so clear in my head because I lived it, and I'm just gonna move on. But thank you. The other benefit of the mussel cages is the amazing animals that like to shelter in there. I'm a sucker for a nudie. Yes.
And how many sites are mussels in Phytoplankton?
So we have 100 sentinel sites. 68 of those are seasonal, and a give or take, you know, some fall away and some get reestablished. And then for phytoplankton monitoring sites, what is it? Something like 80? Does that sound right? No? Close to that. Plus there's people like me that just go sample and, you know, don't have a dot. We're just checking it out. But yeah, over 100 mussel sites. It's a good question. I should have put up the map, but I thought I had 20 minutes, so I was really cramming it. Yes.
So I'm curious with the perennia potential on the coast, are you, since numbers have been usually pretty low, have you seen anything in like the things you're seeing that indicate that ASP might become more in the future?
For the inland waters?
Yeah, or just.
Not necessarily, but that massive bloom Anthony talked about for domoic acid that spanned, you know, California to Alaska, essentially, that was correlated with the marine heat wave, the first really big one. And then when we get mini heat waves, we definitely see more toxin production. It's hard to say if it will get worse in the future, other than it seems like Australis may have also showed up during that same period. So now we have a better toxin producer. So, you know, we have some of the greatest researchers in the world trying to figure out why and when they produce toxin. So hopefully we'll keep knowing more in the future. I mean, we worked pretty hard last year to try to align things in Puget Sound. And I mean, I wouldn't say that we're statisticians, but we certainly didn't find any correlation other than you see something crazy, millions of cells, we're definitely going to want to test shellfish. Plus, as soon as the mussels show up, any DA, domoic acid in Puget Sound, really got cred. Something's different. Yeah.
I wanted to ask your thoughts on any situations you've seen, what it looks like when capacity for shellfish sampling becomes limited in an area. That's something that we've seen kind of locally and regionally. The beaches happen to be closed simply because there hasn't been capacity to go out and sample shellfish, so we don't know. So that's something you've seen, and your thoughts on messaging to the public, because we have worries of, okay, if people see that the beaches are consistently closed, then they're gonna start ignoring that messaging.
They do. I would say that in Washington, only recently have we had that issue where it was a capacity thing, but it's in a toxin hotspot. The messaging does get hard. People get really frustrated, and in particular, the Whidbey Basin for butter and varnish clams, anywhere where there's butter clams. And you also have PSP, people get really, really bummed out. But in 2022, that same thing happened, and then somebody got sick a year after that bloom happened. There's a reason that we're doing it. I wish that there was a way to just make people believe that this was a thing and to pay attention, but if someone does have an issue, you can actually always send them to Jerry and I, because if they're butter clam diggers, we're probably going to ask them to send in a sample for us. We love those connections. It actually really helps with county staff, our staff time. There's all kinds of great reasons to have somebody connected to their beach and their result. But generally that's just for butter and varnish clams. Like, we're really adaptive when it comes to active blooms. I hope that helped. Okay. Yeah, just tell them to check the map and call. So, thank you.
Thanks, Tracie. I'm up next. All right, just full of logistics here. Share screen. There we go. Okay. Hi everyone, me again. So I'm just gonna do a quick overview of some of the data you all collected last year. That says 2023. Let me make sure these are the right slides and that I just didn't mess up the, yeah, I just didn't update that slide. So that's all right. Here we go. From 2024. Oh wait, it's not shared. Okay, share screen again. Apologies. Okay. Okay, so last year, just some quick stats, 29 sites were reporting for SoundToxins, but that totaled to 968 individual sampling visit events, which also was 9,969 individual taxa observations. And there were 80 individual or like unique taxa reported. And I think that number is just for phytoplankton, not counting your unknowns and not counting some of the zooplankton that you occasionally report as well. So even more than that, but 80 phytoplankton. And 114 alerts by email, you guys saying, hey, we saw like a high number on this day and chose to email us. And this is separate from when you put it into the map and it lights up automatically, but for like new things that you're like, oh, it's the first Alexandrian we're seeing this year. And things like that. And then from that, we passed 30 of them on to Department of Health. And so great work. And again, I did not update that, but this is for 2024. I did update the slide, 2024. So just some regional climate trends that were observed last year were that there was a pretty significant El Nino event last winter, so like 23 into 24, which led to kind of warmer winter conditions. And then two things that really affect the Puget Sound include the upwelling timing on the coast and also the river discharges. And those two things interact to really affect kind of what nutrients and are available for phytoplankton, as well as like stratification and just movement throughout Puget Sound. So overall last year was a bit drier than normal. So there was a record low discharge from the Fraser River, which brings, I think, like 60% of water into the Salish Sea. So that's when to pay attention to, but each individual river that drains into like smaller basins also matters as well. So for example, the Skokomish had a higher than usual discharge, so Hood Canal was behaving slightly different than the rest of the sound. Also, the upwelling season occurred like a month earlier last year and lasted 2 months longer. So in the end, started a month earlier and ended a month later than usual. And these are just things to like kind of think about and consider when we like are looking at our data. I haven't done like a super deep dive into how that's connected to all the data you see. These are just kind of observations I've heard. So one of the things we do with the data you all collect is there's a Marine Waters Working Group that's a bunch of monitors and researchers in the region that get together and talk about the marine conditions every 2 months or so. And so we have folks that have buoys and are monitoring all these different things. And they share like updates, and then I'll share the data that this group collects with them as well. And sometimes when weird things happen, we kind of try and connect and figure out what's going on. So these are just some observations that I got from like that group. Then now I'll share our data that you all collected. So we have air temperature, water temperature on the top, and then salinity. And this is just like all of your sites just thrown up on a plot. Some anomalies in like air and water temperature can be like situational. We also have, for salinity, we usually have around the same area, but when you have certain sites that have a really close freshwater discharge or there's recent rain, you're getting your salinity from the surface water. So there's some lower points on that as well. But I don't think I had any like significant observations on here. I did finish these slides last night, so I didn't have time to really think about it that hard. So one other thing that we can look at is the ratio of diatoms to dinoflagellates. And last year we had this like really nice pattern of, I don't know if you call it nice, but this kind of curve of dinoflagellates. So dinoflagellates are in the red, diatoms are in the yellow. Diatoms are most of what we usually see, but they tend to like more mixed waters, whereas dinoflagellates like stratified waters and warmer waters. And so seeing them peak kind of in the July, August time period usually like makes sense. But then this year, I feel like there's not really that pattern. It was just kind of like a weird year. And this might be somewhat related to some of the regional patterns we saw. So lower river discharge and kind of a change in the upwelling season might have kind of canceled each other out just because you're getting, or the timing of the river and the upwelling matters for like what's happening in the sound. So yeah, I don't have like a perfect explanation for this, but kind of I plotted this and I was like, that's kind of, it's not as pretty as last year. It's kind of like a weird pattern. Okay. Also in others and includes ciliates and flagellates, and then silica flagellates are in the green. Okay, here is just a total of all the blooms that were reported last year. Again, sorry, I didn't update any of the years. This is for 2024. So at the top, the numbers are just how many, I think how many visits occurred in those months. So you know that in January or like November to February, we are going out less frequently. So we're probably reporting less blooms partially because of that, but also partially because there's fewer blooms in the water, but that kind of contributes to the like really low winter observations. The colors are colored according to, I tried to order them. So the blue to green is all of our diatoms, and then the like lightest pink to darker pinkish red are dinoflagellates, and then the really dark reds are like other category, which this year included euglenoid blooms, mesodynium blooms, and tintinid blooms. So yeah, just absorb that for a few minutes. It's hard with this many things to kind of match the colors, but hopefully that gives you a pretty good idea of like what was blooming and what categories. Okay. To just look at our toxic species, as Tracie mentioned just now, Alexandrium tends to peak in the kind of July to August timeframe. That's definitely what we see in this plot. So light blue is anything, any presence at all reported. Darker blue is if it was common, and blooms are in orange. And so this is the number of observations that we observed. And again, this one, we still want to, report it and pay attention to it, even if it's just present, because even lower cell counts can still have a lot of toxicity. For Pseudonychia, we do see a lot of this at our sampling sites, even though it's not often toxic. But this year, it seems like we kind of saw the densest amounts in May, April to June, or actually March to June. But kind of present all year round in lower numbers. And then another little bloom in November this year too, which is interesting. And then here is Dinophysis. Again, we definitely see this one all year round, little bit more in the summer, but then also definitely some blooms in the winter time, especially in bud and lit, again this year. And then I've also shown our Shellfish Killers this year. So I think for the past couple of years now, the middle column Phaeocystis, we just haven't been seeing that much of it, which is good. We don't want too much of that one around. For Akashiwa, which we all got to look at yesterday, which was fun. Definitely a late end of the year, July through December, really. We were even seeing high numbers in November and December of this one. And then for Protoceratium reticulatum, again, I think not like the heaviest year we've had in the past couple of years, but a couple of blooms in July through August, and a little bit in April as well, which is also interesting. And actually, I think that's all I have for you today. This was supposed to be my thank you slide, but it is not here. But thank you all so much, because this is all the work and that you all collect. And also, I want to say thank you so much for your patience in this past year, just as I've been trying to manage everything, but we're really happy to have some new capacity and help this year, and hope to do some more cool stuff going forward. So thanks, and I'll take any questions.
Last spring's bloom emerge, should this be the standard?
There's definitely more than 29 SoundToxin sites. Which SoundToxin sites?
There's 29, there's more than 29 sites. Some of them are opportunistic. These are like our regularly reporting, like weekly sites that are currently monitored. And that's the other thing, we've had, folks kind of drop off and trying to get a couple people back on. So this is just like who reported in the past year. And yeah, this is just in the SoundToxins program. That doesn't include ORHAB sites, just to clarify. And as far as who also does mussel sampling, I think we've often talked about how when you do phytoplankton sampling and mussel sampling at the same site, that can be really powerful. But I don't actually know how many do both. But we've been talking about like connecting folks more to make sure the mussel collectors and the phytoplankton collectors are in contact. Yeah. Gonna add to that.
Yeah, Rick, I'd love to, maybe if you can pop on, but last spring's bloom was really, really a lot. One of the other really hard parts is that there are only mussels on the jetties on the coast. So you've got razor clams and mussels, that's it. And then mussels are on the Columbia River jetty and then the Grace Harbor jetties. So that's a really big expanse of beach. So, and really dangerous to get those coastal mussels as well, right? Timing the tides, hopefully not falling down the rocks. It's really tricky. The few samples that we did get last year were a labor of love, that's for sure. We've even talked about testing the razor clam gut, kind of to let you know new bloom's coming, but it really went, the cell counts went from something like 4,000 cells per liter to 35,000 cells per liter in about a day and a half. But Micah with Fish and Wildlife on the South Coast did so much emergency sampling. It's really hard to say how we could get better at this, other than we've actually decreased our commercial sampling threshold from 80 micrograms in the mussels to 40. So we're actually doing that sampling right now, and it's February. But that's just what we're seeing, and kind of to adapt to that. Just for some context too, we haven't had a commercial closure on the outer coast for oysters or anything other than razor clams, and that's just domoic acid, since 1997. So we had a lot of years to think that we had it handled, and then it just shocked everybody. But Washington still did really, really well, and for us to be able to weather that with nobody getting sick is really incredible, and is really a testament to the work that Micah did, state parks jumping in, and even industry just really working hard to get samples to the lab, which is no easy feat from Long Beach to Shoreline. So I really think that the only thing we can do is maybe get some boats out there. Work with the satellites. We got a lot of that on the horizon. So
Anthony's light fish, which we'll probably talk about in his talk later,
yeah.
also getting additional samples off the coast,
Mhm.
which could be helpful to initiation sites specifically, where they know, what is the eddy called?
Oh, Wanda Fuca.
Wanda Fuca eddy. and other hotspots out off the coast that could be indicators. So
Yeah, the other thing, Rick, that makes it so interesting is in Puget Sound, in the inland sea, we deal with Alexandrium and PSP all the time. It's just all the time. You're seeing it everywhere. The second you see a cell, something's happening, right? We're seeing it accumulate in shellfish immediately. On the outer coast, because it hadn't really been dealt with since 1997, it really took a lot of people by surprise. And that's industry, that's harvesters, that's all. I can't speak for Oregon, but it certainly seemed that way. It threw everybody for a loop, and we learned a lot from it, and there's still more to learn.
is this related to this question? Or is it
different question?
Okay, do you mind if I just jump in with one more thing? The other thing I should say is that, the 29 is just talking about SoundToxins specifically, but if you go online and look at our maps, we're also seeing all of the ORHAB sites and also the Oregon sites report on our database as well. And one thing that was unique about the spring PSP event is that the Oregon's HAB sampler was one person who does all of their HABs monitor or like analysis for all, I think 8 sites along the Oregon coast. They were in between HAB specialists. The previous one had left and they hadn't hired the new one. I think she started in like June, which was like right after the event. So she came into like a big thing. But that's kind of also, we've talked about kind of one of those missing pieces, which was part of the reason why it caught Oregon by surprise, was they didn't have HAB sampling for like a couple of months, just at the absolute wrong time, unfortunately. But now they have that sampling again. And so, and also that ORHAB and Oregon and SoundToxins are all like connected, and we're trying to get more connected as well. But we're all, using the same database that DOH is using. So if stuff starts lighting up in Oregon in the future, they'll know about it, like we know about it. It's connected in that way. So hopefully that helps. Okay, you had another question.
Say a little bit more about how drier conditions and potentially record low river discharges could be impacting numbers that we're seeing.
Yes, great question. So my partner is actually a physical oceanographer, and I was picking his brain last night because I was like, I want to know more about this in case this comes up. So let me try and do what I can, but also maybe we can toss it over to Cheryl or someone else in the room that has more knowledge. But the river discharges are bringing in fresh water, and of course the ocean water that comes in is salty. And there are nutrients coming in from the ocean. That's a huge source of nutrients. We also have anthropogenic nutrients from the land that impact. But when the salt and the fresh water meet, you can have stratification. And that also can play a role in like what phytoplankton you're seeing and what they're exposed to, especially at different sites and different times before things get mixed up. So let me try and remember what he was saying. So though the upwelling season started a month early, there was also record low river discharge. And so they might have kind of canceled each other out because you weren't. All right, hang on. We're not entirely sure exactly like what effect that had, but they're both very important in what was observed. Are there any oceanographers in the room that want to help? Cheryl? Thanks, Cheryl.
That was pretty good. I'm a physical oceanographer. Hi, I'm Cheryl Greengrove from UW-Tacoma. And so the way it works is the fresh water comes in, it's lots less dense. The salty water comes in from the bottom, just like you described, very, very dense. It sets up this stratification like your salad dressing that isn't mixed up yet, right? So that layer acts as a barrier for phytoplankton to actually access nutrients at the bottom. So typically high stratification tends to favor dinoflagellates because they have little flagella and they can't move very well, but they can actually move up and down in the water column and access those nutrients. Whereas the diatoms are floating at the surface, they can't access those nutrients very well. So often you will see higher stratification waters favoring dinoflagellates, okay? So I noticed you showed in the 2023-2024 diagram that we saw typically you get the diatoms in the spring, you get the dinos in the fall, right? That's why the stratification is setting up. And if you look at this year, the dinos, their abundance was pretty consistent throughout the year, probably due to that drier setup and more stratification generally throughout Puget Sound. So I think that actually speaks to exactly what you said. We're seeing it in the phytoplankton picture. Okay, so that's just a really quick synopsis of what we think happened, right?
Yeah.
So.
Thanks, Cheryl. Any other questions? Okay, awesome. Anthony, you're back in the room. Does that mean you're ready to go?
Sure.
Okay, Anthony is up next. I did change the agenda a little bit. And so there was a little bit of a confusion, but if Anthony's ready to go, we will have him go next. And then after that, we'll have a quick coffee break for the folks in the room. And then we'll follow that up with our virtual presenters from the EPA and from Woods Hole, who I think are already online. So hopefully we can get those going right after the break. So
You have a stick.
can you join the Zoom? Yeah. Okay, join the Zoom and do it on your computer.
No. Thank you.
And then if you are able to join the Zoom, you can put the sound on yours and the video, then people will be able to see you.
Yeah. Thanks. Actually,
I think if you start the presentation first, you can share just the shared presentation.
okay. So, slideshow. Except for now, I can't.
Can you like? There you go. Okay, I'll download it. Share. So now you can share. Wait, X. Try again. Share. This one. Share. Okay. All right, I think we're good to go.
Okay.
All right, awesome. Thank you.
Thank you.
Everyone.
Hello. I think I've seen most of you yesterday. And I think there's most of the folks that are online have attended some of my Zoom trainings. Anthony Odell with the University of Washington Olympic Natural Resources Center. I'm stationed out in Forks, Washington. I have been with the ORHAB partnership since its inception back in 2000. And I work with all these wonderful people on the outer coast. Quite a big group. It's quite a collaboration. And I'm going to kind of talk about how it was formed and the work that I have done over the last 25 years briefly. Maybe. Okay. So this is a little bit about me. I started off as a fish and wildlife technician, just doing HAB sampling on the South Coast. In 2023, they were looking to transfer from a federally funded program to a state funded program. So I started working at University of Washington and took on a role as a coordinator and a trainer. There was kind of a lot of turnover with the tribal staff at the time because it was soft money grants. And so people would kind of come in, take the job, and then find a full-time job and move to that. And so we had a lot of turnover. In 2025, they did switch over to a state-run program. And in 2007, they gave me a boost up to research analyst lead. And I kind of officially became the coastal sampling coordinator and event response coordinator at that time. In 2008, they sent me to Denmark, where I attended the Intergovernmental Oceans Commission course on harmful algae identification. If anybody has an opportunity, there's a US HAB course, which is really, really good. The IOC course is also, it's especially good. And you have kind of also a very large group of international instructors. It's a 3-week course, but there's 9 months of online training as well, reading and things. If you have an opportunity like this, it's pretty solid HAB course. In 2019, I attended the US HAB course. You know, kind of a refresher. I kind of thought I was going to go in there and I knew all this stuff and it was going to be fine. And what did I learn the hard way? That a lot of things had changed. And there was a lot of updates and, you know, it was really good for me as well. If you have the chance to attend that course, you should. I think there's a few people in here that may have. And in 2024, this last fall, I was really, really fortunate to go to the advanced phytoplankton course in Naples, Italy, which I've told some of you about. It started in 1974. It only happens about every 4 or 5 years. It's a pretty small select group. There's researchers that go, but it's generally people that do HAB monitoring. They want people that have a kind of a broad idea of phytoplankton overall and not specialists. The average age of the people in the course was my age, 52. So that was actually kind of cool. We really nerded out of finally, you know, I found my tribe. The phytoplankton jokes were like nonstop. We had specialists come in, a group of specialists covering dinoflagellates, diatoms, flagellates, and there'd be like 4 instructors and they would go through their talks. We'd look on the slide. It was super intense. It was super intense. It was like 8 to 10 hours every day for 3.5 weeks. Jakob Larsen came in one morning and he's like the dinoflagellate guy. And he's like, okay, good morning.
today we will cover 1,000 dinoflagellates. And we all kind of laugh like, "Ha, you're so funny." He's like, "No, really prepare. We are covering 1,000 dinoflagellates." Like, okay. So really great course. The resources that I brought home and the connections I made is actually the most valuable part. A lot of my talks I've used, gone back and they're like, I can't share those talks, but I can take all the information from all of that. So I'm kind of still in the process of absorbing that. Over the course of the last several years, since 2008, as you guys have known, you've all been a part of my taxonomy courses, just workshops. I've well over 20. And just within the ORHAB program, I've trained well over 25 managers and HAB specialists through the course of the years. Most of those have been tribal folks. And it's been a really cool job. It's been a really strange progression. I don't have my master's or PhD. So I started out as a tech and I worked my up to where I am. In the course of the Naval's course, I was the only one without a PhD. So I thought that felt actually kind of cool. On the ships or in NOAA or the Coast Guard, they call that a Mustang, where you start on the deck and you work yourself up to the bridge. So I'll never be like the captain, but I have quite a bit of experience doing this. The program itself, it started in 1999. There's all kinds of pictures of me training people. There's with Joe Schumacher and Rita Horner, who trained me in phytoplankton taxonomy the first 5 years and on. But with the pilot program, it was her job to train all the technicians. I had no idea what an incredible rock star Rita was when I met her. I had kind of a vague idea of phytoplankton in general. The first course that we had, she provided us a notebook like a month before. I had it memorized for that first workshop. So when she was going through, I'm like sitting, 8-hour course. I'm like, this is so exciting. I was lucky. She really took me under my wing. She's a bit harsh, but I learned. And I didn't realize what a big deal she was until we went to the 2003 US HAB course in Woods Hole. And I was just walking around and she was like, "Come on, Anthony, let's go tear apart some of these posters." It's like, okay. So we're kind of walking around and, looking at stuff with her. Then people come up to me and ask, "Are you like Rita's grad student? Are you like a postdoc working with Rita?" And I was like, "No, I work with her." "You work with her?" And I told her what I did. And they're like, "You are so lucky." And I was like, "Am I?" I really, I had no clue. Truly, everywhere I go, like when I went to the course in Naples, I told them who I worked with and who trained me, and everyone went like, "Rita Horner, like the Rita Horner." I'm like, "Yeah, no, I was that lucky." But you'll see a lot of people like, here's me and Vera Trainer. So again, right off the bat, I had no clue who these people were. Vera gave me her CV because she wanted me to update mine. When I looked at her CV, I was just like, who is this person? She has done so much. Well over 200 publications, started off in PSP, switched to kind of pseudonychia, but she does everything. And yeah, now she's my immediate supervisor, which is it's just wild to me that I got kind of struck by lightning, right place at the right time. And then, yeah, there's several different groups. It's kind of evolved through the years. It's me as a Fish and Wildlife Tech. I'm so young. So yeah, in 2000, it was a federally funded program. This is kind of the original group that started. We had kind of some more researchers involved. Noah was heavily involved, did a lot of training. We talked about Brian Bill a lot, right? And that's the guy who trained me in all my lab stuff, all my field sampling. At sea sampling, near shore sampling, it's been Brian Bill. Like he's a friend, but he's kind of like a phytoplankton god. Like honestly, like he really, really knows his stuff. Always should be someone, I go to him all the time when I have questions. But yeah, he's trained me a lot. We still work fairly closely together. In 2005, we switched to a state-run program, and there's politics between the tribes and the state, and so it couldn't be that Fish and Wildlife was going to take it over. That just wasn't going to fly. And so University of Washington Bean Research Institute they thought that might be a little better, better collaboration and coordination. So ONRC, which is a research facility out in Forks, that's for University of Washington, they picked it up, and that's when Vera and Miranda Wecker, who worked for ONRC at the time, came to me and was like, how would you like this job? You've kind of been here the longest, and you seem to have the ability to train folks. So it kind of moved away from research and went into monitoring and harmful algal bloom event response and mitigation. And this is currently what we look like on the outer coast. So, we have the macaw, the quilly, the hoe, the quinault, fish and wildlife, and then myself. I just kind of generally do event response. I try to sample clay lock on a regular basis. It's kind of central coast. It's a really strong upwelling zone. You have a good mix of diatoms and dinoflagellates, and I kind of can catch things on both sides. It's generally more diatoms on the secondary coastline down on the beach. It's like flat sandy beaches, so it's like surf zone diatoms. And on the north, it tends to be kind of more of a mix of dinoflagellates, especially as you start getting up into Nia Bay. And that's because it's primary coastline, so it's headlands and embayments, little pocket beaches, things like that. Dinoflagellates kind of prefer that. They don't like to be disturbed, right? So they like that area up there. Every one of these entities that we work with, unlike your program, which is wonderful citizen science and volunteers, these are all trained HAB specialists. These are positions that they fill. The quinault, and who covers a large section, and fish and wildlife, have a full-time HAB specialist as a paid position. That's the only thing they do. The tribes to the north, like Rio in the back, he works on a lot of projects, so he wears a lot of hats. So this is like part of his job. And they actually kind of carry a heavier load because they have so many things that they have to do too. And then we put out these kind of either annual summaries or monthly summaries, event response reports. I kind of make these pretty maps and write up a summary, show cell counts and toxin levels, and send it out to kind of a larger, broader group. And then we also have Pacific Northwest Harmful Algal Bloom Bulletin. That's Ryan McCabe. He's a physical oceanographer at UW NOAA. He's kind of moved over to NOAA. He puts these together, but it's all of our data that goes into this to help him make that analysis as far as risk of Pseudonychia bloom domoic acid event. So as far as training and outreach, I mean, this is my role. That's what I do. I've been really, really lucky. It's not just folks at the different tribal labs, but also when I worked at had an office in Grays Harbor College. I started a volunteer program, which Tracie was like one of my OG volunteers. Yeah, in 2015. So the big bloom that happened out on the coast. I was actually doing a biology lab with Tracie. Went out to the beach. We're gonna just show them how to sample, take a water sample, go back to the lab, look in the microscope, see if we could identify a few things. It was like this monospecific bloom of Pseudonychia. I'm like, actually, I need to go make a phone call. But yeah, so yeah, she's up in the top. That's our propaganda photo where we're all kind of looking off into space. But that's the 2016 volunteer group there. Really, really great folks, all of them. Yeah, I mean, you probably recognize maybe a few people here, mostly tribal stuff. Emily Bjornsgaard, as she said before, was in my 2018 volunteer program. It's been really cool because, we'll just do talk about protists one day, and I'll, go into the biology lab. The biology professors already have these volunteers kind of picked out for me. Like, I think she might be really interested, or he might be really interested. So I was like, okay. So you give the talk and then be like, oh, so this weekend or in a few weeks, I'm going to put on a phytoplankton workshop. It's usually spring, so it's like May. So they're giving up a nice, summer weekend to come in and listen to about phytoplankton for 2 8-hour days. The people that can stay awake through that and still seem like they're interested, I'm like, hey, would you like to volunteer for me? And then those folks, if they really seem passionate about it, I try to hook them up with NOAA so that they become a NOAA volunteer, so they get some sea time. And we have various pictures. I think you might recognize Tracie in a couple of there. But we go, this is the Shimada, but I've been going out on kind of NOAA ships and university ships since I think 2003. But it was really after the 2015 bloom that I tried to get volunteers, and we piggyback with the hake survey. So they worked this really great grid doing a stock assessment for hake, which is bottom fish, where they start about, well, lots of times they'll leave out of San Diego or San Francisco, but they go down to Santa Barbara, they start at the coast and they work offshore, and they go about 10 nautical miles and they do it again. So we can sample HABs all the way up the coast and get a really big picture look. A lot of those folks have gone on to careers. I've taken people out of nursing, out of accounting, out of forestry, out of veterinary science. And as soon as they find phytoplankton, they're like, you know what? This is actually kind of cool. Yeah, Becky Smoke is another one. She actually was my Oregon counterpart and led the Oregon HAB monitoring program. And now she's doing scallop research in Rhode Island. So, and she was going to work, she wanted to be a horse doctor. So, but yeah, it's kind of like me. When I saw my first dinoflagellate, like, it's over. Like, I have to figure out what's going on here. So. One of the other things that's kind of new is this is our autonomous surface vehicle. We've done 3 missions so far, but this is the first one off of the Washington coast going out to the Wanafuka Eddy. Those are the places we sample. I'm kind of the main operator, but I'm also training folks how to do this as well. I really need operators and people to learn how to do this because this thing goes 24 hours a day. I can't. The mission is 48 hours. There's prep time. You have to have eyes on it. It's not, some people have this idea that you put it in the water and then it just goes and does its thing. No, this is a $250,000 vehicle, and there's ships and there's debris and there's all kinds of things. It's really smart. It has object avoidance, it has AIS avoidance, so it avoids other ships. When it makes a move to avoid a ship, most of the boats are like, what's that? And they go after it. And then the AI just keeps kind of running away, but then, yeah. You have to take over control basically, but you have to watch it the whole time. This was by far our longest mission. John Mickett is the PI. He works for the Applied Physics Lab at University of Washington. And when we were out towards the end, I was like, come on, just one more sample station. He's like, Anthony, like, we have to think battery life. And, it's really far out there. I'm like, come on, just one. And then he's like, okay, I'll give you one. But we really push it to the max. I think we came back in with like 16% battery life. So it is solar powered. It can run indefinitely. One of these is going from San Diego to Hawaii to Japan, back to Hawaii, and it's between Hawaii and San Diego right now. If you go about 1.5 to 2 knots, and you have decent sun, even at our latitude, it'll go indefinitely. If you want to do things in a hurry, it's going to draw down that power. It can go about 100 nautical miles on just the battery that it has. If you had no solar power, some of these do have a methanol generator. We took ours out to put in a water sampler. So we have a hose that goes down into the keel that samples at about 1 meter depth. We've done some work, but now we're getting like really good samples. Even naked flagellates are coming up in there and they're not disturbed, which that's kind of impressive. We get that back in it's like a compartment full of bags. We can also filter samples at sea. So we worry about the length of time it takes to get back. As soon as you put something in a bag or a bottle, it starts changing like immediately, right? You have grazers, you have bacteria. So on the outside ones, we will try to sample onto a filter, and so there's filtering rig inside this as well. And then as we get closer, we try to do bags of whole water. It's really cool. This is our second autonomous surface vehicle. The first one we spent 3 years on, and it just wasn't going to work. It was very complicated. It had a sail and it could dive underwater. Had a submarine hull. It didn't do good in the surface. There was all kinds of problems. Some of those engineers from that company started CSATs that made this, and their motto is boats that work, which kind of a shot across the bow, literally, of the company they worked for before. But they do. This thing, that whole kind of area on the top provides flotation as well. And they've put it in the surf down at La Jolla, like going over the falls of a wave. and they had an aerial drone kind of watching it, and you're like, oh man, and it just pops, it rights itself, and it heads back out to its loader point. And so, and also, yeah, you can, the user interface, you can just poke on the map, go to here, go to here, go to here, go to here, line them all up, and then say, take this much water at this place, do this here. It has an onboard fluorometer as well, and so you can read that in real time. It has backup comms, satellite uplink. When you go across the patch and it starts showing you high chlorophyll, like actually, let's stop and let's sample here. It's such a great tool. It does cut into my sea time though. It means I'm doing less small boat work and big ship work because, you know, I have this now. We partnered with OSU and NOAA as well, and this is the original one. It's going down to Newport. And the brand new updated one is at APL right now, and they're putting in all the new updated stuff with John and I and their group have talked a lot about what we really need. So I'm actually getting the 2.0, which I'm really excited about. It's going to be housed shortly, April. Yeah, so that's where we are. We started off with what, 2 samplers sampling 4 sites in the surf zone. And now we, you know, it was like the Quinault tribe, the Fish and Wildlife, you know, just really getting an idea. A lot of research went on, like the Barb Hickey's group. HAB was like a kind of a sister research program. They're figuring out basic oceanography. Now we move to all of these groups. We have pretty much all of the coastal tribes, except for maybe the shoalwater. We're working on that, involved, who sample regularly, twice weekly, up and down the coast. We have this really good picture. We work with the Olympic National Marine Sanctuary. They take samples offshore. You know, we're working with other researchers, and now we're to the point where we're using autonomous surface vehicles to go out and do the sampling for way offshore. So yeah, it's come a long ways in 25 years. So this is the hard slide. This is Icelandic. Krefa og takk fyrir. It means farewell and thank you. So I am leaving University of Washington, and I have some really good opportunities in Iceland that I'm following up on. And I will be doing HAB monitoring. I'll still be in phytoplankton. I went there for 3 weeks, and I met some folks in the Naples course. Absolutely fell in love. So yeah, this is on the north side of the island. They have obligations to the EU and the other Scandinavian countries, and they don't have the people to fill those positions. It's a very small island, and so they're heavily recruiting, and they're like, now's a really good time to recruit Americans. They made a very good offer, and we're working on that position. But I met the director, I met their phytoplankton person there, went to school there in Naples. And they want someone to kind of do HAB monitoring in the more remote areas during the summer. And then in the winter, I'll be doing fjord sampling off a ship. So which is like, it's ideal for me. I love the climate. As you know, I'm like always sitting next to an open window, right? I was there and I was like in a t-shirt the whole time. They're like, oh, you Icelandic? And I'm like, no, I just like, I'm hot all the time. So yeah, and some of you know too, I'm a polyglot, so I'm actively learning Icelandic, which also it's difficult, but it's fun. Yeah, thanks. Any questions? Yeah.
Can you remind us who funded the glider?
The glider?
Yeah.
Oh, so that is money through NANUS and Jan Newton. And that went through APL. And yeah, and that's kind of who's started that. It's going to be transitioned over to us. I'm sure APL will be involved still. So.
And then the filter on it is just for nucleic acid.
We can do anything really. So we had to increase the pore size because of it would clog up, and then we're kind of stuck there. So it's slightly bigger. So I think a lot of smaller flagellates go through, and I also think some of the smaller pseudonychia probably go capture pretty much everything that's out there. So yeah.
Big thanks to NANUS.
Yeah, huge things. Yeah, yeah. Thank you, Jen. Thank you so much. Yes.
Can you leave us with a phytoplankton joke?
Oh, man. They're really spontaneous. So yeah, I'll think about it. Maybe I'll write some up. I'm still going to work with Michelle to, I don't leave UW until March 30th. And so a lot of you have done those Zoom trainings. We recorded those, but I'd like to make some more, like Thalassocera versus Cosmodiscus, stuff like that. All the things that form long chains, how you can tell the difference. All the little round brown things, the lurbetes that you can confuse for Alexandrium, Ursos, unrecognized, what is it, round objects, yes, which are like all those very small flagellates, right? That, you know, unknown flagellate. There's a lot that we can work on there. You can actually, there's a lot of these things you can tell what they are on a light microscope if you're looking at live samples. So we're still going to work on putting some of those talks together and giving some more taxonomy training, but then you will have those. So yeah.
Nula versus Lepori-Bui.
What's that?
Nula versus Lepori-Bui.
Oh yeah, all of those. Yeah, absolutely. Yeah. Anyone else?
Okay, next up, we've got Naomi presenting from the EPA, and Naomi, you can feel free to introduce yourself and go ahead and try to talk now, and we'll see if everyone can hear you.
Okay, can you hear me?
Try one more time.
Can you hear me?
Good in the room? Okay, you're good.
All right, I'll get started. So good morning, and thank you everyone for giving us a slot in your agenda. I'm going to switch gears a little bit and talk a little bit about an EPA project, Saving Puget Sound from Eutrophication, Understanding Nutrient and Cyanotoxin Hotspots, and Finding the Best of the Best Management Practices. I'm with EPA's Office of Research and Development. I'm actually out of the Atlantic Coastal Environmental Sciences Division in Narragansett, Rhode Island. We have a diverse team. Stephanie Anderson is a marine phytoplankton ecologist. Steve Rigo is a coastal ecologist. Avery Tatters specializes in harmful algal blooms and associated toxins, both in marine and freshwater systems. And Craig Connolly is a coastal biogeochemist, and Tori Walters is our GIS specialist. So I'm just going to spend a little bit of time giving you a broad overview of our project and focusing in on the second component, which is dealing more with HABs. I'm required to present this disclaimer. I also want to acknowledge our EPA Region 10 collaborators on the project, Sydney Clark, who's also on the call, Rochelle Labiosa, and Rory O'Rourke. So as you all probably know, excess nutrients are a major stressor in Puget Sound, and there's interest in finding the most cost-effective ways to manage those nutrients to meet loading targets, as they can contribute to both HAB problems as well as low dissolved oxygen. So there's interest in tools to optimize nutrient reduction, and that's one of the areas of focus of our overall project. A second is HABs. Changing conditions in the watershed, temperature, increased storminess, population growth are likely to exacerbate nutrient inputs and conditions favorable for HAB development. And we all know there are associated threats to both human and animal health associated with the algal and cyanobacterial toxins. A fair bit is known about the marine biotoxins in Puget Sound. Less is known about potential problems with cyanobacteria, and we're hoping to improve our understanding of the spatial and temporal dynamics of cyanobacteria and algae, and also help to identify most appropriate upstream management practices to address nutrients in the system. So there are two research questions to our overall project. The first is looking at the most cost-effective management strategies to reduce nutrient loads to targets identified by stakeholders in the basin, and how those strategies might be modified to adapt to changing climate, and how to incorporate margins of safety to address differences in weather over time. The second, which I'll focus on here, are looking at the combination of factors that are the best predictors of the probability of algal blooms in particular focusing on cyanobacterial component in Puget Sound. Where and when do we expect risk to be greatest? So as you all know, since you're heavily engaged in monitoring, the historic focus on toxic algae in Puget Sound is centered on marine algal species, Alexandrium, Dinophysis, and Pseudonychia. Whereas in freshwater lakes, the focus has been on toxic cyanobacteria blooms. Some of you probably heard these historically referred to as blue-green algae. They're actually bacteria. Researchers have found evidence for bioaccumulation of microcystin toxins from cyanobacteria in shellfish in the coastal Puget Sound, downstream of lakes with cyanobacteria blooms. But in other coastal systems, cyanobacteria have found to be an emerging problem in estuaries and freshwater tidal rivers. So it's not necessarily just a problem from downstream transport. There are conditions under which you can get cyanobacteria actually growing in coastal systems. So these are the different components of the overall project. We're developing chlorophyll time series for Puget Sound. There are a variety of measurement platforms out there to monitor chlorophyll in the sound, but they don't provide continuous spatial and temporal coverage. So we're using satellite imagery. I'll talk a little bit more about that to develop a time series. We're pulling in information on nitrogen loads from the USGS Sparrow model for the basin. USGS just released a dynamic Sparrow model that provides information on not just average annual loads, but now seasonal loads over a 20-year period, so we can look at historic changes. We're also considering other driving factors that influence both algal blooms in general, as well as the dominance of different species, things like temperature, discharge, nutrient loads, stratification, and retention time. And then tying that with ground truth sampling at potential HAB hotspot locations and trying to work with partners in the area on that aspect. So we have, we're relying both on Sentinel-3 and Sentinel-2 satellite imagery. Sentinel-3, there's an example shown here that's actually from our friends to the north, who post these images online. The satellite 3 imagery is a little bit coarser, 300 meter pixels, but for many parts of the sound, that's sufficient. It's also the overpasses are more frequent than for Sentinel-2, about a 2-day interval for each of the 2 Sentinel-3 satellites. Previously, there are problems with using Sentinel-3 satellites for coastal systems because you have more turbidity from suspended sediment and dissolved organic matter that can interfere with the signal for chlorophyll. But there's a new chlorophyll algorithm that's been added to their data that's good for optically complex coastal waters. So we're using that particular indicator. We're also working with Sentinel-2, has a finer resolution, about 10 meter pixels. So you can get more detail along the shoreline. It does have the disadvantage that it requires more pre-processing in-house. There's some new improved methods to mask out clouds and cloud shadows and do better atmospheric corrections for Sentinel-2. And we're in the process of going through that to provide historic satellite images, process images for Puget Sound. So with the Sentinel-3 data, we've done an initial hotspot analysis to provide information to inform sampling priorities. We've also done some initial analysis of the outputs from the dynamic Sparrow model that just got released to the public. So we're looking at a total nitrogen exposure index that takes into consideration both loads from upland sources, from marine discharges, and from atmospheric deposition, rainfall directly on the surface of the water, as well as looking at the temporal and spatial distribution of chlorophyll as a measure of algal biomass. So this shows a map of our exposure index for total nitrogen from the 2014 output from the dynamic Sparrow model. And again, we've taken the Sparrow outputs and supplemented them with atmospheric deposition directly to the Puget Sound surface, as well as the marine point discharges from wastewater treatment plants and other industries. This map doesn't include the Strait of Juan de Fuca because we have incomplete data on loads for that system. And we've broken it down by quarter of the year. Here, the green represents the highest effective concentrations, and the dark blue represents the lowest concentrations. So I'll just cycle through the different quarters. The pattern stays pretty much the same with the highest exposure from inland sources in southern portions. The southern Puget Sound and Hood Canal, also the most northerly portion, but some differences across seasons, about slightly more than twice as high exposures in Q1 and Q4, so the wet season, basically, as you might expect. We've also looked at the probability of hotspots based on exceedances of various chlorophyll thresholds. 3 different thresholds. This is the highest one we looked at. We looked at 23 and 68 milligrams per cubic meter thresholds. These have been associated with higher probability of toxins from freshwater algae in freshwater systems. And we also looked at an intermediate threshold of about 41, which is 5% above the median for chlorophyll overall in the system. So the red represents hotspots where there's a greater probability of chlorophyll values exceeding those thresholds from 2018 through 2024 during the growing season, May through November. The blue represent areas with a very low probability of exceedance, and the gray represent areas with no significant tendencies either way. There are some differences between years, but overall, again, you can see higher probabilities of exceedances in the southern portion of the sound, as well as a swath across the Strait of Juan de Fuca. You will note that Bellingham Bay does not have a hotspot on this map. The hotspots for Bellingham Bay actually varied between years, but it's not showing up on this particular graphic for the 68 threshold. I mentioned there are various factors that determine whether or not you have an algal bloom and what species predominate. So we're looking at chlorophyll, nutrient loads, as well as other predictors, things like salinity, temperature, retention time, which magnifies the exposure to nutrients, water column stability, stratification. Just as dinoflagellates do better in stratified waters because they can move up and down in the water column, the same is true of some cyanobacteria species. We don't have, from the Sparrow model, we don't have measures of nitrogen inputs from the ocean, which can be important at times, so we'll be incorporating information from the upwelling index for that aspect. These are some of the questions we're interested in looking at toxins from freshwater versus marine species. What's the distribution of algal toxins such as microcystins from freshwater algal species across Puget Sound? Are these associated with high chlorophyll levels and or high algal toxin levels in upstream lakes? Or are they also associated with high chlorophyll levels in the subestuaries within the Puget Sound? And what other conditions might be associated with their production? We're hoping to coordinate with existing monitoring. As I showed previously, there's a potentially higher risk in the southern portion of the system with high nutrient loads, relatively high nutrient loads, and also some bloom hotspots for algae in general. These are also some important shellfish areas. Shown in brown here are the DOH sentry mussel sites, I believe. we're in the process of finalizing our sampling strategy, but currently we're considering sampling both in spring and fall, as well as a finer temporal sequence off of Seattle near the EPA Region 10 lab. The sampling will consist of both phytoplankton sampling as well as with IDs, as well as putting out spats to look at both marine and freshwater toxin presence absence. So with that, I'll leave you with the contact information, and I'm afraid I didn't keep track of time. Hopefully there's a minute or two for questions.
Thank you. Yeah, I think we do have a minute or two for questions if anyone has any. See if anyone's online has any questions. No? All right, awesome. Well, oh, wait, we have one.
Systems in mussels in Puget Sound. What are the hotspots for detecting mussels with cyanotoxins or microsystems? Have you found any?
Could you hear that question okay?
Yes, we haven't started our sampling yet. We're only a few months into this project. There have been a few studies that have found bioaccumulation of cyanotoxins in mussels, mainly in the southern portion of the sound, and those were near outlets of tributaries connected to lakes that had high cyanotoxin production.
There's also a question in the chat about if you monitor silica as one of the nutrients.
We should be able to include that when we're doing the grab samples for phytoplankton. Unfortunately, the Sparrow model doesn't predict silica. There is some ongoing monitoring in Puget Sound that hopefully we could pull some data from the actual monitoring water quality data.
Any last questions? Okay. Thank you so much, Naomi, for that presentation. Nour, are you online and are you ready to go?
Yes.
Awesome. Hi. Welcome. Yes, and do you have permission to share? Oh, perfect.
So,
okay.
Can you see my slides?
Yeah, but we can see like it's not started the slideshow. It's like the PowerPoint screen.
Is it good
Yep.
now?
Yep, perfect.
Great.
Okay, thanks. Take it away.
Thank you. Hi, everyone. I think it's still morning for you. My name is Nour Ayache, and I'm a research associate at Woods Hole Oceanographic Institution at HUI, working with Dr. Mike Brosnahan. And today I'm going to present you the work that has been done with the Shellfish Killers project, led by Dr. Vera Trainer, and the title is Effects of Emerging Harmful Algal Toxins, especially Yesotoxin, Spectrin-toxin 2, and Azaspiracid on Pacific oyster larvae. So this study was done last year with Dr. Vera and Dr. Misty Peacock at the Shellfish Sea Research Center. Sorry. So just for a brief introduction, the US ranked third globally in bivalve consumption, with Washington State ranking second for aquaculture production. So this shellfish, sorry for this. Oh, yeah. This shellfish industry is really important for for the economy because it's provide job and income for thousands of farmers and businesses in the area, but also because it holds a cultural importance as a source of subsistence and ceremonial food for Native American communities in this region. And we know that from the last few decades, there have been a lot of efforts from both federal and state agencies to promote the growth and sustainability and prevent depletion of oyster beds in the area. And this was done by promoting larval and seed shellfish production in hatcheries, But despite these efforts, there have been a lot of like challenges like production failures or mass shellfish mortality events. And historically, we know that these mortalities were linked, for example, to bacterial or ciliate contamination or poor water quality or acidification. But recently, there have been a new threat, which is the presence of HAB species and their associated toxins near shellfish production areas. So some of these emerging threats or HAB toxins are Pectinotoxin 2, Yesotoxin, and azaspiracids. And these toxins have been recently detected infiltrated into treated hatchery water on the East Coast, for example, or present or detected in like near key shellfish cultivation area in Washington State, for example. And we know that these toxins, like Pectinotoxin 2 and Yesotoxins, are not regulated here in the US, but recent studies, like field studies and lab studies, have been shown that Yesotoxin and Pectinotoxin 2 have been linked to shellfish mortality events, or have from lab studies, we've seen that these toxins had negative impact on the healthy development of some shellfish like oysters and scallops, especially in their early life stages. But so far, we don't have any studies regarding the impact of azaspiracid on the shellfish growth or survival or fitness. So for this study, we decided to test the impact of 5 different toxins: Pectinotoxin 2, Yesotoxin, azaspiracid 1, 2, and 59, and expose the oyster larvae. They are 20-day-old pedivaliger larvae, CG gas, to these toxins. And we did 3 different tests. So in the first test, we exposed these larvae to a range of concentration of each of these toxins individually. For example, for Pectinotoxin, it went from 1 to 100, Yesotoxin the same, and for azaspiracid, it went from 0.01 to 1 nanogram per milliliter. But we also did a combination of these toxins together, and we exposed them for 24 hours. In the second test, we did the same process, but we added adrenaline to induce their metamorphosis or their settlements. And in the third test, so test number 1 was up to 24 hours, they were rinsed and put into a clean medium. And then we added adrenaline to test the recovery. So just to mimic, for example, if a bloom was passing near a hatchery and infiltrated, or toxins that were infiltrated into the hatchery, but it was cleaned after 24 hours. So just to test how the larvae will survive or behave. So after these 24 hours, we measured the larval mortality and metamorphosis using microscopic observations, and we were able to calculate the LD50 and EC50. So EC50 is the effective concentration that led to 50% of the larvae to settle or metamorphose. Okay, so in the first test, when we looked at the effect of these toxins on the larval mortality, so these graphs represent the like percent of mortality for each tested toxin. So we have Pectinotoxin 2 in green, Yesotoxin in orange, azaspiracid 1, 2, and 59 in different shades of blue, and a mix of these toxins together. And as you can see, we saw an increase in larval mortality with the increase of the concentration of each of the tested toxin. And we also were able to find, for example, the first toxin concentration that caused significant mortality compared to the control treatment. And it was like it's shown here in the red arrows. So it's 5 nanogram for Pectinotoxin 2, 1 nanogram per milliliter for Yesotoxin, 0.5 nanogram per milliliter for AZA1 and 2, and 0.75 nanogram for AZA59. We were also able to find the maximum effective concentration, after which we had like a saturation in mortality, and it's shown here in the green arrows. And also, when we combined these toxins together, we found a significant higher and additive effect on the oyster mortality, and the highest mortality was found when all these toxins were combined together. Next, when we looked at the effect of these toxins on the larval settlement or metamorphosis, we saw a similar pattern. So when we increased the toxin concentration for each of the tested toxin, we saw a gradual decline in the settlement, and we were for each like toxin also. I'm sorry. And we were also able to find the first concentration that had like significant impact on their settlement. And when we combined the toxin together, we saw also a higher decline in the settlement rate or percentage. So from this data, we were able to calculate the LD50 and EC50. So these plots represent the dose response curves for each of the toxins, and the plot on the left represent the mortality, and on the right, the metamorphosis. So as you can see, and as I mentioned before, we saw a gradual increase in larval mortality with the increase of the concentration of each toxin, and a gradual decline in the metamorphosis or their ability to settle with the increase of the concentration of each toxin. And from these graphs, we were able to calculate the LD50 and EC50 for each toxin. And as you can see, we couldn't calculate the LD50 for Cyanotoxin because we needed higher concentration, higher even than 100 nanogram per milliliter, which is already like a significant dose. So from these results, we were able to conclude that AZA1 and 2 were the most toxic toxin on the oyster larvae survival and metamorphosis, followed by AZA59, and then Pectinotoxin 2, and the least toxic was Cyanotoxin. Next, when we looked at the effect of these toxins on the recovery, here I'm going to show only the example of Cyanotoxin. So as I mentioned before, the larvae were exposed to each different toxin. We left them for 24 hours. Then we rinsed them, we put them into clean media, and then added adrenaline to induce the settlement. So here we can see for the control treatment, for example, after 24 hours, the larvae will start to develop a foot. And then when we add adrenaline, they lose this foot, they develop gills, and we can see a significant shell growth. However, in the Cyanotoxin treatment, even at a concentration that caused only 10% of mortality, we didn't see this normal development. And even if the larvae did not die, but they didn't recover properly and they did not progress into a healthy juvenile. So we know that these toxins, the Pectinotoxin 2, Cyanotoxin, and azaspiracid, are present in Washington state coastal waters, and especially during spring and summer season, which is coincided with the natural and spawning and early development stages of oysters in this area. And from this study, we showed that these toxins had lethal and sublethal effects on the development of oysters because it impeded their growth into settled and healthy juveniles. And we were able also to calculate the toxicity threshold level for each of the tested toxin, and we found like Pectinotoxin 2 is more toxic than Cyanotoxin, and then AZA1 and 2 were more toxic than AZA59. So in conclusion, the presence of these toxins in coastal areas where the hatcheries are present, in addition to other like biotic and abiotic disturbance that could arrive or that could happen also, like increase of temperature and salinity, acidification, or other diseases, could increase the threat of oyster recruitment and production yields, and like have a significant economic and ecological consequences. So for future perspectives, we're hoping or we're planning to repeat this set of experiment with using the Manila clam larvae, and also investigate the long-term effect of these toxins, but using also adult oysters. And if we have like extra time, we're hoping to include other environmental stressors with the HAB toxins, like increase the temperature and salinity, and see how that adult oysters will react to the presence of these toxins. And that's the effect on their ingestion rate, clearance rate, and depuration rate. Thank you so much for your attention. I know I speak very quickly, so I think I used less than 10 minutes. But I wanted to thank the MIRHAB Shellfish Project for funding this study, and especially also Dr. Vera Trainen and Dr. Misty Peacock for allowing me to do this research with them, and for Taylor Shellfish Farms, Jamestown Scallum Tribe Shellfish Farm, and Lumi Bay Shellfish Hatchery for providing the animals. Thank you so much.
Thanks so much, Nour. We have a couple minutes for questions. Does anyone have any questions? Yeah, we have one in the room.
I'd love to get some idea of how these concentrations of the toxins that you're speaking about compares to the cells per liter metric that I think a lot of us who monitor plankton, as relative amateurs generally think about.
Yes. So of course, the range of concentration that were chosen were environmentally relevant, and they were based on some values that were found in the literature, but also from concentration that were, for example, detected into treated hatchery water on the East Coast. We have some data from the West Coast, like from the SoundToxin program. They were lower, so that's why we had to go the extreme, also high concentration, to be able to calculate the LD50. So for Cyanotoxin, I think the concentration above 100 nanogram is really unpredictable to find it, or not yet found in some or documented in papers, but yeah, so the rest of the concentration, especially for azaspiracid and pectinotoxin too, they are really environmentally relevant concentrations. And were chosen from bloom densities that, for example, for Dinophysis ranging from between 1 and 10 cells per liter. So that's why we base this concentration.
Thanks. Any other questions? No? Okay. Thank you so much, Nour. Great to have an update on that project as well. Since we've all been looking for those shellfish killers in our samples for a couple of years now. So that's the other part of the work that's going on with that project. Okay, so our next presentation is going to be Cheryl. And I will pull that up now, and that's the last one before lunch, and then we'll take a lunch break.
Do you have a pointer or a clicker? No.
No, but if you move your mouse,
Okay.
it should go through, and then you can hit this button. Do you like to walk around? Actually, I do have a clicker in my bag. It's just I might have to pull it up. Okay, one
super, because I can't see behind here. I'm too short. If not, I'll do it the old-fashioned way. That worked really well. Want to thank Michelle for the tech work she's doing today. This is not easy because nothing talks to anything else and you need adapters and.
Should automatically turn itself on, but here, hang on, let's stop the share real quick.
It's not on. It's not, you can't see the.
There's no laser. Oh, I hope the batteries didn't die.
I bet it did.
Oh,
that's all right. Okay, we'll do it another way.
Okay.
Yeah.
Bummer, I should have checked that before.
It's okay.
Okay,
Good.
we'll try one more. We'll get this back up there.
Right.
Oh, it's two
It's
triple A's.
It's triple
Oh,
A's,
but
and
thank
I
you.
don't have any in my, I usually have lots of things in my backpack that I don't have those.
If it helps, I can
No, it's okay. I'll do
Okay.
it.
All right. Share. That's the front.
Yeah, it might be helpful to someone later in the day.
Yeah, triple A.
Yeah.
Okay.
Okay, can everybody online see okay?
I think so, because we're seeing it up there.
Okay.
Okay, so you should be good, and then arrow keys just
Okay.
right, left.
So, hi, everyone. I'm not used to standing behind the podium. You can't see me. But I will do that today. So welcome. We're gonna give you a little tour of a new method we've developed for mapping the distribution of Alexandrium catenella cysts in Puget Sound. I'm going to give you a brief history of what we've done in the past. Some of you have seen part of this before. I've been here before at SoundToxins. And the front part of this talk is basically a repeat. If any of you were at the Shellfish Growers meeting last week, I gave this there as well. Slightly different angle, but the same thing. And then the second half of this is I'm working with part of the Marine Waters group that Michelle mentioned earlier to put together a phytoplankton vital sign indicator for Puget Sound. And this is, the vital sign indicators are run by the Puget Sound Partnership. And we've been working, 2023, we've been working and had a bunch of workshops around phytoplankton. Some of you probably attended those. I know you have. I've seen some of you there. And we did a follow-up grant with Puget Sound Partnership funding us to put together a database. And I think this will tie to Naomi's satellite data. So Naomi, I hope you're still online. And then we're actually moving forward with that database. And we'd like SoundToxins' help in informing that database. Tracie's provided some from Department of Health, and all the agencies in the area and some of the tribes have also contributed data to that. And I'll tell you about that. So I have two separate talks that Michelle asked me to talk about in half an hour. So here we go. So as you know, many of you here are experts in your local areas. Alexandrium has been around for a very long time. This is from Vera's paper on the right that shows we have an increase in the occurrence of paralytic shellfish toxins in the shellfish. And that's from Vera's paper back in 2003. But we know it's been here for a long time. We actually see it down core from studies. And there are records in George Vancouver, who actually mapped Puget Sound for the first time from the European perspective, that some of his crew actually died on that trip. And it's documented in his journal. So it's been here a long time. It's nothing new, but we do see more of it. And as you know, part of the life cycle for many dinoflagellates is they overwinter in the sediments here in Puget Sound. It's a way to survive many, many years. And then when conditions are right, like typically as it warms up here in Puget Sound, these little cysts will germinate and become swimming vegetative cells. And that's when they can be filter fed by shellfish, right? So that's when we watch for Alex in our samples for SoundToxins. And when we really start monitoring more heavily for the paralytic shellfish toxins with Department of Health in Washington State. And there's Alex in chains in the middle. And these little cysts, these are not to scale on here, but these little cysts, I'm afraid to touch this. I don't know if this is going to work. Yeah, I don't have, like, Max and me are not happy campers. Okay, so there it is. This little, I'm going to give up on that. Okay.
Do you want me to point?
No,
to
no,
the
it's all right.
timeout?
It's cool. I'll just say
Okay.
where it is. So these like, what do we call them? Jelly beans. The one on the left is actually what it looks like under the microscope, no staining. But what we do is we actually stain them. So when we want to see how much are in the sediment, right, we can actually count them more easily because they stain this bright, like Julie calls them, little yellow jelly beans, right? So obviously we're concerned about this because it can cause paralytic shellfish poisoning. So all that stuff, you know. So what do? We, over the past few years, we've actually gone out and mapped. You have to go out in the winter because that's where in the sediment and they're not moving around so much, right? We go out in the winter, which is lovely conditions, and sample the sediments in Puget Sound. And, you know, originally we started this project with Rita Horner and Jim Postal at UW. Our first mapping of the sediment cysts in Puget Sound occurred in 2005. We went out again with Stephanie Moore and Vera Trainer, part of this project. We went out in 2011, 2012, and you can't see there 2013. And collected cysts and maps where the cysts were in Puget Sound. And as I said, it's a very time-consuming process. You have to go out in the winter, collect the sediments, then you have to basically sonicate the sediments to get all the junk off the cysts, sieve them, preserve them. This takes multiple days to kind of etch the outer. They're very resistant outer, we'll call them shells. and you etch them so that then when you stain them, the stain takes to the outer shell and you can see the little yellow jelly beans, right? This is a documented method by Yamaguchi that's been used for years, since the 90s. And then Julie, who's below me there on the microscope, sits there in the dark with fluorescence and counts them and listens to, I don't know, Alex in Chains or something. So these are the result of those Puget Sound wide survey maps of cysts in the sediment. And from 2005 through 2013, the times we got out, these were all EcoHab funded. And you'll see the most recent project is MareHab funded by NOAA. And you can see the predominant hotspots stay fairly consistent. The hottest spot often is Quartermaster Harbor there on the bottom of those maps. And then also Bellingham Bay tends to have a lot of Alex. And then the little bays on the western side of the main basin, where you see that 500 there, those tend to be warm also. And the little bays like Swim or Discovery Bay often will get hot as well. And so you see a trend here, a lot of the little bays, not the main basins or the main channels, the deep channels, because they're too deep. The little bays tend to be great little incubator bays, and a lot of you sample is really helpful. So those cysts, if you kind of look where the hot spots are for the cysts, you can see, and this is just summarized data for the whole year for 2011, 2012, 2013. This is the paralytic shellfish toxin data from Washington State Department of Health. And you can see that the hot spots where we end up getting hot shellfish is basically where the cysts are, pretty much. So getting these cysts and knowing the relative abundances can be really helpful as an early warning system to people, shellfish growers yourselves included in the area. And so with the cyst and the SoundToxin monitoring, we can both inform Washington State Department of Health and help with human health in Washington State. We also have gone out and done emergency response mapping for Alexandrium, where there was a big bloom and they're like, okay, where do they end up? The shellfish growers want to know, right? The recreational harvesters want to know. And so the first one we did here happened, as you know, the blob came in 2014. Everything was warmer. In fact, we thought there was something wrong with our instruments because it was so warm. And then we all talked to each other and were like, oh, it's like real. This is weird. And then we got phone calls from the papers that says what's going on. And Nick Bond said, it's the blob. And then it became the blob forever after Nick Bond named it that. Anyway, so the blob came in. We had this huge bloom of paralytic shellfish toxins in Daybob and Quilcene Bays up on that little hook, the little thumb of the mitten of Hood Canal. And we were like, uh-oh, we typically don't see it in there. And so shellfish growers approached us and said, can you go out and sample where the cyst ended up? So this is just, and this is a little tiny maps, but it's 14 on the bottom, 15. So you're going up in time, right? The blob came in 2014, and anywhere where you see red dots is where that's the shellfish toxin on the left. And then the two maps on the right are cysts per centimeter in the sediment. So we had the bloom in Daybob and Quilcene in 2014. We went out that winter in January and did sampling and found lots of toxins, lots of cysts where they had found toxins in the water column. We found cysts in the sediment. We didn't find it much further south. We found a little bit by Seebeck, right? But then the following year, we said, okay, we've got lots of cysts. We presented at the shellfish growers meeting and said, you guys need to watch more carefully. SoundToxins was there watching more carefully. We saw Alex in the water column in April and basically put out an early warning to Jerry and Tracie. Were you there yet? You weren't there yet. It was just Jerry at that time. And so they actually sent shellfish down and they shut down the shellfish bed. In fact, they stopped trucks going to market, right? So a very effective system jointly between monitoring cysts, SoundToxins, and Washington State Department of Ecology. You should all be really proud about the kind of collaboration we have here in Puget Sound. So this is an example of how we can use it for an early warning system. You heard from Tracie this morning about how we had a big Alexandrian bloom on the coast in Lepori-Bui and Grays Harbor. We went out 2 weeks ago on that little fan boat with Fish and Wildlife. That's me and Julie on the boat with Zach from Fish and Wildlife, and sampled the sediments in Lepori-Bui where we had a weather window. We're still waiting to get a weather window for Grays Harbor, because now, of course, it went to heck, I'll say that. And so we have to get out in a small boat and do the sediment sampling in Grays Harbor. We'll do that in the next 2 weeks. And the map on the left is the one that Tracie showed you this morning, where the hotspots were in the shellfish paralytic, paralytic shellfish toxins in the shellfish on the left. And then we went out and collected 31 sediment samples, which right now I have a student in the lab processing that. And then Julie will count those in early March. And we'll have some idea of where the cysts ended up in Lepori-Bui. And we'll do the same for Grays Harbor. So again, one of these emergency response type mapping that can help us help the shellfish growers prepare, right? We're also working with some modelers. Terang and his postdoc have just got a model running for Lepori-Bui. Neil Banas had done a model in the past about flushing rates, and we'll give some of our data to them as well. What else do I want to say? Oh, and so this is all, both emergency response mapping is funded by Abrica as part of the emergency response funding through NOAA. And so they approached us at the last HABs meetings and said, you know, Jerry said, can you go out and map? And Zach said, I have a boat. And NOAA said, we can give you a little bit of money. Can you do it? And that's how it happened in November and why we're sampling in January, right? So these things can happen very quickly. And we thank NOAA for that. All right, so the project that I started my topic on, it was like finding a new method to do this. Obviously, it takes a long time to count all those cysts, right? Is there a quicker way to do it? And so we teamed up with the NOAA Beaufort Lab. I want to combine all my words today, BOAs. Steve Kibler and Mark Vandersea to actually see if we can use a qPCR method, looking at the DNA, actually the RNA inside the little cysts. And can we get an idea of abundance that way by feeding it through a machine rather than Julie looking at a microscope, right? So that's kind of our goal here. And so we got a ORHAB funded project, and this actually spanned 3 areas. We wanted to know if it worked everywhere. We want to know Gulf of Maine, because they have a big sampling going on there, right? Cyst sampling. And they're just starting cyst sampling actually over the past couple of years. Alaska's gotten really hot, and that's a big issue up there because there's a lot of subsistence food that people rely on. And then here in Puget Sound. So we said, okay, let's do it in 3 areas. And so what happens is we collected sediment samples for like 3 years in those 3 locations, sent them all to Julie to make sure they're counted right and make sure the counting, that the labs counting with microscopes are consistent, which I can tell you they are. That's a good thing, right? So like training our eyes. And then we sent all the samples that we collected, a split of those, to NOAA Beaufort Lab to see if they could do the qPCR technique. Then we'll compare that with the eyeballs of the microscope counters, right? So that's kind of our game plan. Here's sort of the maps real quick. And, you know, you don't have to look at it, but the one on the top is Gulf of Maine. The one on the right is Puget Sound. You see our same hotspots, Bellingham Bay, Quartermaster Harbor, and those little western bays in the main basin. And then the bottom was our Kodiak samples from Alaska. I will tell you, there are more cysts in Alaska than there are anywhere else. So be aware, the actual volume is bigger. And they're seeing huge blooms in Alaska now that it's warming. And that's a real concern. So, so we went out and mapped those. Looks good. Now, let's take a look at our method and see how well we did. So if you look at the graph, but then I'll walk away from this thing. So if you look at the graph on the left-hand side, it's qPCR versus manual count. So qPCR, thank you. qPCR is the y-axis, the manual counts are the x-axis, And so you can see you have a pretty good correlation. This is just using the example from Gulf of Maine. And then what we did is map them both side by side, one using our manual counts and the other using qPCR. In the case of Gulf of Maine, it overestimates the actual amount, but the pattern is the same. And it's the pattern we want to see, because then they feed it into a model that predicts where the floaty cells, right, the vegetative cells are going to end up, right? And so they use it as they have a coastal predictive model for this. It's a lot harder to do a model. We've tried a bunch in Puget Sound with Stephanie Moore and Neil Banis. We're a lot more complicated system in Puget Sound than this nice, smooth, open coast, right? So it's a little harder for us to do that sort of modeling, but we are working on it. So we basically run our samples through. I'll show you a close up of this. Okay, so what we do for the qPCR is we take the sediment, we filter it, much like we're prepping for the microscope, but then we put it in this bead beater, because these things are so resistant. We had to find an instrument that could actually open them up and get the DNA out, right? That was a big part of it. And then getting the right kits, and then we fed it through the qPCR. Now, as part of this process, we have to do a standard curve, right? Which says, okay, what the qPCR is telling me, how many cysts does that really represent, right? So if you look on the left, increasing DNA is down, because a qPCR it makes copies, right? So the more cysts you have with this stuff in it, right? The more cysts with this stuff in it, the less copies you have to make, okay? So it reads sort of upside down, which is why I put these big arrows there. It's like, it's like your upside down graph. So your increasing number is to the right on a standard curve. And the amount of DNA is actually lower number of copies for this little machine to make, okay? So keep that in mind. And so we went and we did standard curves for each region. And then we said, okay, let's see how our manual counts stack up with our qPCR counts, all right? So that graph on the left is the graph you saw in the example I gave. And then that was for October 2020. We did it again in February 2022. We didn't get out 2021 because this is all COVID, right? And we did it for Puget Sound. We have a pretty good fit there as well. Kodiak needs a little more work, and they're up there again this year, working on that on a follow-on project. But this method does work pretty well, right? Doesn't give us the exact amounts, but it gives us the same trends, which is what we're looking for. But when we develop these standard curves, so there's our little standard curve up in the upper right again, where we use like little cysts mean, this many qPCR cycles means this many cysts. When we look at our standard curves, we're like, I wonder how they compare with vegetative cells, the swimming cells, the cysts. And if you look at the one on the left, they stack up pretty well. Vegetative cells and cysts, really the gene copies and our standard curve looks the same. But when we plotted our standard curves for 3 different regions, they're different. You can see Gulf of Maine's on the top, Puget Sound's in the middle, Kodiak's on the bottom in that middle graph. So they're not quite the same. And so I said, I wonder if there's a difference seasonally, depending on when we pick up the cysts, right? Maybe something's going on there. And so I went to Quartermaster Harbor, where we have lots of cysts. Thank you, Quartermaster Harbor, for being a test site for the rest of the planet. And we went out, Julie and I went out every 2 months, collected sediment, shipped it to Beaufort. They ran a standard curve, and you can see there's a shift seasonally on when you pick up the cysts. So what this tells us is we can use this method, but every time we thought we could just develop one standard curve for the planet. It changes depending on where you are. It changes on the time of year you're doing it. So you need to basically create a standard curve every time you go out and do a survey. So how does this all stack up? Well, it works. We've shown that, but we need these standard curves, right? And we know the qPCR saves time. It's less time intensive than Julie sitting at the microscope counting little yellow jelly beans. And, but we still need to set up that standard curve, which is an extra thing. And if you want to get this up and running, let's say we want to do it in Washington State with Tracie and Jerry, we would have to set up the lab with this instrumentation up front and train people. And it's most, like, if we just want to go out and do like a small bay, like Grays Harbor or something, it's not, it doesn't save us that much time. It's when you have lots of samples, like if you wanted to monitor on a regular basis, 100 stations, then it's worth your time to do it, right? So that's what we found out. We can, as we said, future for Puget Sound, we can use it as an early warning tool, as we have for the large surveys. We can augment existing monitoring like I talked about, and we can use it for emergency response mapping, right? That kind of thing. So my information's there. That's the website with all of this project in it. So now I am going to switch gears. And we're going to go from cysts in the sediment to phytoplankton writ large floating around in Puget Sound. Okay. So this is another thing that I do. So some of you were at the phytoplankton workshops we hosted as part of the Puget Sound project. Amy, were you there? I think you were there for one of them. Tracie, you were there, right? Yeah. So we ran these workshops in between 22 and 23. A bunch of us got together from the Marine Waters group that Michelle talked about. And we said we were really lacking a coordinated effort on phytoplankton in Puget Sound, because we should have a vital sign for Puget Sound. We have vital signs for other things, but they're the base of the food web. Everything else depends on the phytoplankton. It's really important that we do that. So we got people together and said, okay, what's all the data in Puget Sound? What do we have? That was our phase one, with the idea that we wanted to pull that together in some sort of a database. So then we could build some sort of a metric or a couple metrics for Puget Sound to get at that sort of vital sign. So it gets it in the public eye, it gets it in sort of the, what do we want? The policy makers' eyes, right? And that's where you guys come in too. So the team is run by myself, Jude Apple up at Purdue Bay, Brandon Sackman, who used to be at Department of Ecology, now he's working for GSI. He is a total data guru, and we're thankful for him because he built most of this. And then Ali Chase, who's at UW APL. Some of you may know her. She's also a phytoplankton queen. She's dynamite. She does a lot of the statistical analysis for us. And then we have a whole science advisory team from various places: Jan Newton, Terry King, Gabrielle Hannock, even though it says King County here, she's retired and she's still doing this as a citizen like you guys. And we have Neil and others from the tribes, Francesca. So that's kind of our team. And this was phase one. We did those workshops. We're in phase two now, towards the end of it. Phase two will end in June. We're going to apply for a new grant to actually develop based on what we pulled together, because right now we're starting to see some really cool data that all of you will be able to use, which is chlorophyll and phytoplankton and things like that. And so then we will propose a metric and some monitoring requirements and requests to the state, to Puget Sound Partnership, right? Which is what we'd like to see for SoundToxins. This was from phase one, where everybody just we had a Google map where people could upload their metadata. What data do I have? You didn't have to upload the data, just tell me what you have in terms of place and time. So we assembled this map in phase one. Phase two part of the project is we're gonna go out and get all that data, right? This was Brandon, it was amazing, and his team. And then we're gonna have an inventory of what we have. We'll do some statistical analysis. This is where Ali comes in to look for spatial and temporal patterns. And then we're going to make some recommendations about this. So we got like 16 regional providers, 40 independent data sets from a variety of sources. I think we actually have more than that now. This is early on. And then the kinds of data we got were, these are some of the places. This isn't the whole list. I just wanted to show you, like Department of Ecology, University of Washington, the tribes, Departments of Fisheries, Oceans Canada gave us some data. And these were profile stations like CTDs, which have fluorometers on them, which will give us an indication of how much phytoplankton's in the water. This is a shout out to Naomi with satellites. We collected mooring data from various locations. And then also supporting data, for instance, here, the Washington State Department of Health, biotoxin programs. We have your data in this database, Tracie. And so we also have some phytoplankton community data from King County. We would like to include SoundToxin data in this if you are willing. And so that's going to be my pitch at the end with Michelle. And so what we've done, Brandon set up this database where we have these areas. So we can show the whole area at once, or we can pull out by those little green boxes. So we can pull out, because we know that phytoplankton don't all bloom at the same time around Puget Sound. Whidbey behaves different than Hood Canal, right? So we need to be able to look at the big picture, but also basin by basin. So we kept basin by basin indicators. This is just some idea. We have data back from Washington Department of Ecology earlier, but we started in 1999 because Christopher Krem says that's where we start having good data. So we're going good data, right? And so we started in 1999 and we have collected the years 99 to 2023 right now. So that's the amount of time we have. The kinds of, that's the spatial and temporal distribution, the kinds of maps we can get out of this. For instance, the upper two maps on the left is just how much CTD, mooring, and bottle data, right? CTDs on the left, fluorometers, the right is bottle chlorophyll data. Tells you how many samples we have. It goes up to like, a couple thousand in each of those. The bottom one shows us how much chlorophyll we have, samples by season, in different, by different data sets. So there's King County. Let's see, I think we got one from one of the tribes and the other one is the ecology, right? And then the graph on the right is just 2023 to 2016. You can't quite read it there. But it shows you in all the basins how much data we have and what the data sets are. So we have all this ingested. We can make all these plots that we want. And actually it's designed so you can go in and look at the data too, which is part of the advantage of how Brandon has set this up. We've set it so citizen scientists can go in and look at chlorophyll data if they want by basin. And if scientists want to use it, they can download the data and he provides the actual code in Python and R. And that's all on GitHub for those of you that may be programmers. You can get the code off there and plot it any way you want. So the idea is to make these data accessible to everyone, to recreational shellfish growers, all the way to scientists that may be looking for other patterns that can help us, right? So that's the game plan. Hopefully I can do this. I want to show it to you live. Oh, okay. I need help. I need assistance. I am not. Yeah. Okay. So
You're
click on
trying to-
A.
I want to change the
Oh,
screen. I'm
can you
sharing.
just share overall? No, you have a Mac. I'm not gonna. All right.
Um,
So can you unshare and then reshare
yeah.
that? Because this is the website. It's live on GitHub.
Do you
Okay,
want me to
so I can.
navigate here at all, or do you want to?
Yeah, I'm gonna have you do it.
Okay.
Okay, so if you slide down, it brings you into this page as a starter. Just keep going down. And you can see the coverage we have, right? And so then go back up across the top. If you click on inventory. And again, this was Brandon just put this all up 2 weeks ago, so it's not done yet. So you're seeing things like beta live, right? And so this is all the data that we got. These are all the entities that contributed to this. And what kinds of data they have, sampling locations by area, and you can also click on any one of these and get information. So click on one on the left. You can click on the sites. You can also click on the left to get a particular area if you want, and it will tell you how much bottle data, chlorophyll data, when it comes from, where it comes from, and all of that there live. And you can also download these data. He also put up some distributions about where this is, like where are your samples, from what time. We're starting to put up these heat maps in here. And I'll show you some ones that Ollie put together. So if you go back up to the top, there's, I think, data management. There's another thing there that talks about that. So if you click on one of the guys on the left, let's say, I don't know, pick one of those. Sure, pick that one. I don't know what do we got? So it tells you about the data source. It tells you the information about where the data came from, what it includes. So everyone has access to everything. And what we're going to do is I'm going to show you some of the products. We're going to have standard products that you can look at by basin. You'll be able to pick by basin and look at products. So let's get out of here because you can click on analysis. There's nothing in there yet. But I can show you some of the graphs that Ollie put together, which I have on my PowerPoint. So if you go back to the PowerPoint.
Okay.
So be aware, once this is totally compiled, we will send it to you. You can go in and play with it, okay? By your basin area to look at chlorophyll, which is probably what you're interested in relative to phytoplankton, and if there's any phytoplankton data for your area, right? Which is what we'd like to get. So the kinds of products we're going to see. So if you look at these, we call them heat maps. So 99's on the bottom, it goes up to 2023 on the top. So each of those is a year. And then across the bottom starts January to December. And what this shows us is how many samples we have in any given year, right? In any month. And this is for the whole Puget Sound, but we're going to do this by basin, each of one of those little basins. So you'll know what data is available for your area, right? And then you can go in and you can plot it. So these actually tells you sort of median values of what the chlorophyll is. And we just did some tests. So this is from, I forget what time frame. I think Ollie did like 2013 to 2023. She did 10 years here just as a test. And those are the data sets that we have. The bottle, the chlorophyll median on the left are the bottle sources. The ones on the left is, CTD, and the ones on the right are bottle. And so you can see what kind of data we have available and when it gets hot. One of the things we're very interested in is and this is one of the questions, I love what Naomi did because what she's looking at with satellite data is exactly what we want to propose for qualitative metrics for Puget Sound phytoplankton. We want to look at chlorophyll. When does the bloom start? How long is the growing season, right? And we can get that from these data from chlorophyll. But we also want to know about HABs. So we need community data from people like you to be built in the data set so we can say something about what's there. Not just how much is there, but who's there, right? So, so we have a lot of these sort of plots like this. We're going to build by basin that you will be able to download. You can also look at time series. This happens to be from 99 to 2023. The bottle data and the fluorometers on the CTD data. You can see where we get all the peaks. When we had high years, right? When do they typically start, right? And you could pull this down by your basin, right? So, how can SoundToxins help? So this is the pitch, and Michelle's helping me with this. The database includes chlorophyll data from bottles and fluorometers on CTDs and moorings. We also have phytoplankton community data, for instance, from King County, a bunch of the tribes, and we have the Washington State Department of Health, the SoundToxins programs, Tracie and Jerry's data built in there, right? And if you think about it, there are different kinds of data, right? And so this is where SoundToxins can help. We have the chlorophyll data, which kind of gives us better temporal and spatial coverage, and it's kind of numbers, right? But even there, we're doing sort of a qualitative picture of when things happen, when it's big, like it's not there, it's sort of there, it's really there, right? So you have sort of the stoplight approach, you know, from green to yellow to red, right? Or in our case, maybe it gets greener, I don't know. But it's that sort of qualitative approach of when does the growing season start? How long is the growing season? That's a very qualitative thing, but we can get at it with the chlorophyll. The other part of the metric we need for Puget Sound, and what we're going to tell Puget Sound Partnership they need, is Basically, we need to know something about the phytoplankton community. Because that's what's tied to the food web and to human health when we talk about HABs. And so we have some of that community data, but it's patchy. And you are the main place that covers all of Puget Sound. So we would like to incorporate, if you are willing, and I know this is done on a site-by-site basis, your data into this database and make it part of the development of a phytoplankton vital sign for Puget Sound, and also make the recommendation that the state should support SoundToxins. So anyway, that's that project. I hope you can help. If you are interested, you don't have to send your data in. Just tell Michelle, yeah, I'm good to go. And she's going to compile a list of names. And since all your data is organized nice and neatly, which Brandon's very happy for, he goes, is it all different? And I said, no, it's all organized. He goes, okay, we can do that. So he's prepared to accept that data. And we'd like to do it sometime in March. So let Michelle know if you're interested. I told our guys that I would ask you for your data to be incorporated there, because it's a really critical part that we don't have full coverage for. So, with that, this is our next phase. We're writing the next proposal to actually pull together, finalize the database, pull it together, and put together a metric that we think will be useful for Puget Sound, and recommend that to the powers that be, basically policymakers. So there you go. That's the update. And I know I'm the only person standing between you and lunch. It's about lunchtime. If you don't want to ask questions now, you can find me at lunch, because I know people are hungry.
Thanks so much, Cheryl. Yeah, so we are excited about lunch, but does anyone have any pressing questions at the moment?
Yeah.
As the DNA in those samples increase, the number of cells decrease?
No, so as the machine, the way the qPCR works is it makes copies of DNA. If you have lots of cells and there's lots of DNA, it doesn't need to make as many copies for it to detect it. So if you put a sample in there and it's got lots of DNA, okay, lots of cysts with DNA in it, right? Then it says, oh, okay, I have a lot of cysts. I don't need to make a lot of copies. So if I don't need to make a lot of copies, then I have a lot of DNA. If I have just a little bit of DNA, how it detects it, it makes copies. If I need to make a lot of copies in order to detect it, that means I started with less. So that's how that qPCR machine works. It's like, think of it like a Xerox copier, right? And if you have a lot to begin with, you don't need to make a lot of copies, right? If you only have a little bit to begin with for it to see it, you got to make a lot of copies. So that's why the y-axis is upside down. And it's really hard. Like the first time I looked at that, I'm like, that doesn't make any sense. But then, you kind of have to think in reverse for that. So yeah.
Quick other questions?
Yeah.
We'll go here and then there.
Okay, the core samples, because we stopped doing them because we thought they weren't really necessarily being processed.
The chlorophyll?
Yeah.
Yeah.
Does that mean there's gonna be funding so SoundToxins can keep? I mean, my understanding was that there
Right.
wasn't anybody actually processing this.
Right, because there's no funding for it.
Right, so great question. So I think that so my understanding, for I've been sleuthing myself, is that I think maybe we had a little bit more ability to process samples when we were partnered with NOAA, but then when that breakup happened, we only were able to do some of the sites through some of the research projects, but then just with COVID and Terry leaving and some changes to what we decided to do with our projects, some of those samples didn't get processed. But if we were able to get more regular funding, we would have to figure out. Right.
Because we could, but we stopped.
But,
Yeah.
right, because you actually need, when I go out into chlorophyll, I have students that go in the lab and they have to filter it, run it through a benchtop thermometer, right? So it's time consuming.
And we can't do that at SoundToxins. We would send them somewhere else, but to set up those contracts and stuff, we would need funding.
And if it's frozen in a little bottle or. I'll ask you details.
we can ask, I can give you all the details. Okay.
Yeah, we can chat more about that.
You sort of answered, but there are what can resolve this.
Yeah, it's really just a bottle sample. And then we can do a couple other things. You can freeze it. You can, saute it right away. Or you filter it.
So I
Yeah,
think
or you can filter it.
I'll just maybe just add to that. So I think right now the ask is to look at the data that we've already collected with whatever we are already doing. So we're collecting community composition data, relative abundance data is
Yeah.
the primary thing that they're wanting to add to this picture. And for those sites that we have already chlorophyll data, that like historical data could be used. And then with phase 3, the question is going to be, okay, so like going forward, how can we keep evolving this data to like make this availability for everyone? So we might add chlorophyll to some of the sites, not all of the sites at that point. But really right now the ask is for the historic data that's already been collected, not for like more work yet at this moment.
Right.
And I think the way we're going to do this is we'll send an email follow-up, just especially for anyone who's not here today, kind of summarizing the like what we're asking for and what that would mean. So like at this time, when people ask for our data, we ask on a case-by-case basis, but once this is going to be up there, it'll be like available for everyone to see. So each site can make the decision whether they want that or not. And it'll be a site-by-site basis. So, not everyone has to participate. If you don't want to, you don't have to, but if you do, then it's a really cool opportunity to have your data that you're working on be used in a lot of different ways.
Yeah.
So we'll send an email after this meeting at some point, like either maybe not this week, but next week, kind of explaining that a little bit more and then asking folks to think about it, talk about it, and then opt in if they want. And then we can do that with the historical data.
Right. The most valuable thing for us right now, you can see we have a lot of chlorophyll data, right? And so we would like to have more, of course. That's a future ask. But the thing that's most valuable is the community data and the HABs, because we don't have that everywhere. And so that's where it would be super advantageous to have your who's in the water data, which we have very limited amounts of, right? And that would be really informative to a Puget Sound vital sign for phytoplankton, right? So, yeah. But that's a good idea. I can, we can make that right. We can say, how about if, since these people are doing this, you can do that and see what they say, right? If you don't ask, you don't get. Exactly. Right?
Okay. Hi, everybody. Thank you for having me today. My name is Megan Schulz. I am a graduate student at University of Washington, and I'm also a research assistant at the Salish Sea Research Center out at Northwest Indian College. Dr. Vera Trainer is my advisor at UW, and Dr. Misty Peacock is my boss and advisor at the Salish Sea Research Center. So, as some of you may remember, back in Memorial Day this last year, there was a surprising and massive paralytic shellfish poisoning event along the coast. Now, this was surprising, and it happened, a lot of people got sick. Some people were hospitalized, but no one died from this event. But it led to a reinvigoration of the idea that we need things on site that can be used by shellfish growers, by people who live or work or in more rural areas. Needs to be some sort of a test. And so this led to a rapid response program or project to try to test out some methods and see how accurate they are. So this made national news. There were concerns about shellfish growers needing to recall their product. It closed recreational beaches. It also inhibits people who are culturally, like shellfish is a culturally significant food for them and sustenance harvesting. So many people were impacted by this beyond the people that got sick. Of the people that got sick, these are some responses of how they were able to track the sickness. So the majority of people that were sick got sick from eating mussels. Next up was clams, a small percentage oysters, and no one from crab. This slide is really just to show that while mussels are often the ones that make the news, it does impact other types of shellfish and the people that eat those shellfish. Now, this event was not only fairly surprising, the saxitoxin levels that they reached were extremely elevated. So the Department of Health has a limit of 80 micrograms of saxitoxin equivalent per 100 grams of shellfish meat. Mussels tested went up to 5,500 micrograms per 100 grams of tissue. This shows this didn't all happen overnight. This was a gradual rising in this toxin. And so if there were ways to test more quickly and in the field, decisions can be made based on this information. So I'm going to first go through some testing methods. So the first is what Department of Health uses. It's the mouse bioassay. And while this is the standard, and all of these other tests are going to be based off of these values, it's not really feasible for on-site testing. It's expensive and it's time consuming, and you have to be kind of an expert to do it. However, there are two other types of tests that I'm going to talk about. So the first is an ELISA, enzyme-linked immunosorbent assay. It can give you quantitative results, so you can get actual concentrations, and you can perform many tests at the same time. However, these tests are prone to user error. They are tricky. I've run hundreds of them. They're still tricky and honestly fairly annoying to run. You need specific equipment to read it. And also there is a concern because paralytic shellfish toxins are a suite of toxins. It's not just one type. And there is some cross-reactivity within those types of toxins that may skew your results. Rapid antibody tests are, as you can guess by the name, quick. They give qualitative response, and by that I mean above and below that regulatory limit I talked about. It's a simple procedure, and it is more feasible for on-site testing. However, these are new, or at least these brands are newer, and additional verification needs to be tested to make sure that what results you're getting are accurate. And by accurate, I mean accurate to the Department of Health levels. So with each of these graphs, I put up a blank one first, just to set the scene, and then I'll throw the data up. So on the x-axis, you can see the Department of Health values in micrograms of saxitoxin equivalent for 100 grams of tissue. On the y are the ELISA response. If a point lies in that green box, it means that the ELISA values match the Department of Health values in that they are both under the regulatory limit. If a point is in the red box, it means that the two tests are in agreement that they are above the regulatory limit. The circles are going to be mussels, which is the majority of the samples tested, and the triangles are razor clams. So as you can see here, most of them are in agreement. The one sneaky one is the razor clam that, as you can see, is in the upper left-hand corner. And that would be the one clam that's a cause for concern, because that means the Department of Health has said this clam would be dangerous to eat. But based on the ELISA, it is under that 80 microgram regulatory limit, which that's when a mismatch gets dangerous. However, these numbers aren't bad. Like the majority of them are in matching. But again, the ELISA is really tricky to do in the field. And this set of data was not just from one ELISA test. This was weeks of work and several ELISA kits to get this data. And this is just 28 samples also. So then we have one of the rapid antibody tests that we tested out. It's called AquaBC. It was previously known as Gillette Rapid Test or Scotia. It is a company out of Chile. And this is from their website, some of the tests that they do. This data comes from Christina DeWitt at OSU. So what I've done with this is I've put up a blank with some fake data so we can go over what the points mean before we jump into the real data. So that first little triangle there, that green point means that it's a razor clam. And it means that the AquaBC response would say it's negative for toxins, so it's below that regulatory limit. And that's in a match with the DOH values because of the green background. This other pretend point means that they are in match because the red circle means that AquaBC said it's above the regulatory limit and the Department of Health also says it's above the regulatory limit. So now we're going to switch to some, oh, this is a mismatch because the background is green and the point is red. So now that we've done fake data, I'll show you the real stuff. So this is the AquaBC results. As you can see, the majority of these points are a mismatch. Everything in that green bar, except for those 3 samples, those shellfish are deemed healthy to eat, safe to eat by the Department of Health, but AquaBC's response said that it was above the regulatory limit. While these mismatches are not concerns for safety, this is a concern for a premature closure, premature recall, or sending your samples to the Department of Health for testing when they don't really need to be tested yet. As you can see, some of the mismatches, the Department of Health said that there was no toxin detected. So this is not necessarily a sensitivity issue, this is a testing issue. So then we move to another company that does a similar style of test. The company is called Sensorial, and the product is called PSP Alert. On the right is from their website. On the left are photos I took while running these tests. This is their old reader. It was a prototype. They now have moved on to a larger one that is a screen. You're able to run several tests at the same time. And if you're running a shellfish that maybe hasn't been tested as much, like a gooey duck, you're able to send that data directly to the company and they can work on what's called matrix effects, which is the difference between shellfish in there. Think of a mussel versus a razor clam. One's a little gooier, one's a little chunkier. That's going to be matrix effects. So this is through Sensorial. When you order the kit, you get the reader, the cartridges that you're putting liquid on, every reagent you need, including DI water, which is great if you're in a place where your water is not experimental clean. All you need is a pipetter. Okay, so to start, where Aqua BC had 2 levels of above and below, Sensorial has 3. So they have high, which is also above that 80 microgram regulatory limit. The yellow is above 30 micrograms, and green is anything 0 to 34 micrograms. And the idea behind this is when you're testing your shellfish, you will know as it's approaching that regulatory limit. So here's the data for this. Just look at it for a second. You can see that it's pretty lined up. There is 2, actually, let's switch it here. So this is if we use all 3 levels. However, if we switch it to 2 levels, because remember, medium is also under the regulatory limit, the mismatches aren't as bad. So what we're really looking at here is the 1 yellow point that's above the regulatory limit and the 1 red point that is under. So with that yellow point, what that means, Department of Health has said, this is not a safe clam to eat, but Sensorial has reported it is under that regulatory limit. And that's the more dangerous of a mismatch. The red that is under the regulatory is just the opposite. The Department of Health has said this one's safe to eat. Sensorial has said it is not. So in summary, ELISA has few false positives. Of the 16 negative samples, only 1 of them was a false positive. There are no false negatives. However, there's potential for mismatches near that regulatory limit because that quantitative number is not always the most accurate. Additionally, it is a tricky test to run, especially in the field. Aqua BC had many false positives. Of the 19 samples, there were 16 false positives, but there were no false negatives. Sensorial had few false positives. Of the 19 samples, negative samples, only 1 false positive, and few false negatives. So of the 9 positive samples, only 1 false negative. I put that one in bold because that is the clam that would be dangerous to eat if you just trusted the Sensorial numbers. Overall, rapid antibody tests have the potential to be a useful tool to test shellfish for PSTs in the field or in remote areas. With more testing to confirm accuracy, they could be a tool to help prepare or prevent PSP events like what happened over Memorial Day. What's next? We have more samples now that have also been run by the Department of Health to help confirm how Sensorial is doing. We want to start running samples in triplicate to make sure that they are repeatable. And we want to expand the species that we're testing. Instead of just mussels and razor clams, we also want oysters, gooey ducks, other types of clams like butter clams to make sure that those matrix effects aren't going to affect the outcomes. And then also we want to try Sensorial's new reader and make sure that the accuracy is as the old one was. And thank you to Noah for the funding. And are there any questions?
There's not an industry out there of any kind that doesn't want red light, green light. Okay.
Yeah.
So, and even in the business model.
Of course, yeah.
So I understand the want for simplicity.
Mhm.
Maybe if you're on a boat, as a a commercial fisherman,
Mhm.
that might be one thing. But basically, could you just set a margin, a conservative margin, like what you had on your band there, and say, this is.
I think you could. So because these tests are private companies, we're not really involved with the mystery of what's happening in the reader. in terms of us getting more information. I think the idea is to use the threshold so that anyone who has no science background is able to run this test and get red light, green light, like you're saying. And also I hope too is to give growers or people invested in this, not financially, but they want this product, the chance to be able to do their own initial testing before sending it to the Department of Health, because the Department of Health is getting so many samples where if I'm able to do this test and see that it's red light, we can hold off. We don't have to send any samples in yet. I think that's the main goal of this product or these types of products.
Just a quick introduction. We were asked to repeat any other questions.
Oh, sorry, yes.
And that was basically about if you could just kind of set a different threshold so that it could be just like a red light, green light, like yes or no. And but for the future,
yeah,
we'll answer or repeat the question.
Yes. Anyone else? Okay, thank you. I'll switch to the
awesome. Thank you. We're gonna have some more Megan now.
double feature.
Double feature. But I will switch the PowerPoint presentation for you.
Okay, should we get to go? Awesome. All right, hello again. I'm still Megan Schulz. All of the associations stick. So I'm here on this talk to give an update on a project that is a culmination of everyone you see on screen, the names here, plus more people that are interested in how harmful algae in the Salish Sea is going to be impacted with the projected ocean acidification and climate change, including rising ocean temperatures. So this work is very place focused. I'm gonna go back, that's too distracting. This project is very place focused. It's Salish Sea, it's a project that is from the ocean acidification group as well as the hazardous algal bloom group. And it was a community interest-led project of specifically tribal members, tribal scientists, federal, state, monitoring communities. There is a concern of what toxic algae what's going to happen with toxic algae with these new conditions, these ocean conditions. And there are 3 species that were identified as being species of concern. And their photos are down there. We have Alexandrium. They produce paralytic shellfish toxin that I talked about in the last talk. We have Protoceratium reticulatum, which is a yesotoxin producer, which is a clam killer, and it's what I'm going to be talking about today. And then we have Pseudonychia that produces domoic acid, which is known as a crab killer. So on the left here, the map, you can see the Salish Sea or the Puget Sound plus Salish Sea here. And those are points where algae is being counted by SoundToxin, and there are buoys that are monitoring for ocean acidification and things like temperature and acidification, pH. And so the project goals are trying to figure out how this is going to influence harmful algae in the Salish Sea by focusing on these 3 species. And to do that, we are going to measure the effects of ocean acidification and rising ocean temperatures, both in the field and in the lab. So the first step is to get a baseline field study of pH and HABs. What's going on in the ocean? What's in the Salish Sea? What are the current levels? What are risks that we're looking at? Then we're going into the lab where we're going to start with temperature, nutrients, and pH, and we're going to see what do those have to do with growth rate and toxin production of these 3 species. And we are then going to make it more complicated, but that's more realistic. And we are going to start doing multivariable studies, including temperature, nutrients, and pH, because the ocean is never simple. It's not just temperature rising. It's not just nutrients changing. It's going to be a whole matrix of all these 3 things. So as we are getting more information, we will be able to present something like this stoplight idea, where not only will we be able to say, hey, there's a bloom of this species, but we'll be able to say, and these conditions that we know can lead to a bloom, all of these are the perfect storm, keep your eyes out. So within this project, I'm a graduate student for it within this project. And my first part of the project, it's a puzzle, part of the puzzle piece of the bigger story is how does temperature impact growth rate and toxin production in the dinoflagellate Protoceratium reticulatum? It is a temperate species and it can be found worldwide. It is a yesotoxin producer that is known as a shellfish killer. There have been mass mortality events of shellfish around the world, including South Africa, Spain, Chile, and more locally, the Puget Sound. Terry King's paper in 2021 describes a mortality event in the summers of 2018 and 2019 that was attributed to yesotoxin. This toxin was previously grouped with diuretic shellfish toxins, but it turns out the same species can make these toxins, but this one doesn't really impact humans. And also it's chemically quite a bit different than the others in the diuretic shellfish toxins. My culture or my isolate that I'm using for this project was isolated by Rosa Hunter from a net toe sample from Blaine, Washington, even more north than Bellingham, during the fall of 2023. So to address the first part with a single variable experiment was how does temperature impact growth rate and so specifically the growth rate part. And we're using a temperature gradient bar, which is basically a long incubator with a lid, and it's insulated. There's a water cooling system in it. And the gradient part is that there's differing thickness of metal. So you're able to grow the same type of algae or different, in this case, it's the same type of algae at varying temperatures. So in this case, we spanned it from 10 degrees Celsius to 22 degrees Celsius, which that range is all reasonable within what we've seen in the Salish Sea. And the photo on the left is the photo of my algae growing. And the photo on the right is from Dr. Bill Coughlin, who helped put this together. So we're going to just take a second. This is a graph or a snippet of one of the growth curves that came from these algae at 3 different temperatures. So the blue is 10 degrees, the yellow is 16 degrees Celsius, and the red is 22. This is over time. And what we're looking at, the Y scale is not as important here, but what we're really looking at is the slope of the lines, because the slope of the lines is how you're going to measure growth rate. And so I've highlighted it here. So here's the maximum slope at 10 degrees, maximum slope at 16 degrees, and the maximum slope at 22 degrees. And so visually you can tell they're different, but they're all they're pretty similar. So what we did is I calculated the maximum growth rate. So all of these were grown in triplicate. And so that's why there's a range in maximum growth rates at that point. And as you can see, there is a decent amount of, I've done no statistics on this, so we're going to be talking in general, like generalizations here. There is a bit of a difference between them within the same temperature. And we think that's attributed to the fact that these are grown in very small volumes. And that's when they start getting impacted by available space and competition, where in future tests, we're going to be using higher volumes, and we expect that range to close or at least lessen. But the takeaway here is that the highest temperature has the highest maximum growth rate. So then the next part of the question is, how does temperature impact toxin production of this dinoflagellate? So this is a little diagram of what a toxin sample looks like. So on the left is the culture vial with the whole amount of algae in it. 1 ml is removed to do cell counts. Then 5 ml is filtered, and you keep the filter to look at particulate toxin, which is the toxin that's inside the cells. And then you keep the filtrate to measure the dissolved toxin, which is the toxin that has left the cell and is now just in the liquid, in the seawater. You can combine those two and you get total toxin. So toxin sampling occurred in 2 different sets and on 2 subsequent days. So on the left, you can see the dotted line there for EXP1, that's exponential 1 and exponential 2. They were done 1 day apart, and just mid-exponential, meaning anytime it's still really going up at that angle. And then on the other side, you can see the stationary 1 and stationary 2. And with that, we wanted to know, okay, these algae have stopped growing, or at least not multiplying as quickly anymore. Are they still producing toxin? So this is what that is looking for. So on the bottom left is a photo just to help remember what growth phase is talking about here. All of this data is, or all these toxin samples were analyzed using triple quad LCMS. The purple bar is the particulate toxin, which is in the cell, and the teal bar is the dissolved, which is in the seawater. And this is for 10 degrees. And if you can see on the bottom there, it's sample A, sample B, and sample C. So these are all replicates of the same temperature. So we have 10 degrees there. You can see a very tiny bit of dissolved toxin at the top there. Here's 16 degrees, and here's 22 degrees. And as you can see, again, we have not done statistics, but 10 and 16, generally speaking, look pretty similar. But 22 degrees is where things look a little different. And so what we can confidently say is that the highest temperature produces more dissolved yesotoxin, specifically in the stationary phase. Now, I took those cell counts for a reason. And what we're looking at here is the toxin per cell. And this is in picograms per cell, which is a very small unit. And as you can see, the 10 degrees, which if you remember back here, was kind of at the same with 16, but here it looks higher. And that's because the coldest temperature grew the slowest. So there was less cells, but still a decent amount of toxin. So in this case, it means the 10-degree samples had more, kind of made up for it in toxin per cell. And with toxin per cell, you also only do particulate toxin. You don't do dissolved because it's in the cell. And that's why the 22 degrees looks also much smaller. All right, so to wrap up this preliminary part, a takeaway is that with higher ocean temperatures, this dinoflagellate will likely exhibit higher maximum growth rates and will produce more dissolved yesotoxin, especially during the stationary growth phase. But this is a small piece of a bigger puzzle. So what we've really also shown is that we are able to successfully grow and culture this dinoflagellate from the Salish Sea over all 5 temperatures, over several growth cycles. We are showing that it is producing yesotoxin at all of these temperatures and at each phase, and that we are able to accurately extract that toxin and quantify it, which is going to be building blocks on future studies, which are shown here. So for next steps, we are going to move to large volume experiments where we are adding in other variables like pH, like nutrient availability. And we are going to be picking temperatures based on projected ocean temperatures of the Salish Sea. This increases the realism and also increases the complexity. And we are going to next go to nutrients and temperature before getting into pH. Special thanks to Dr. Bernard Borman, who's my official advisor at UW-CEFS out at ONRC, Rosa Hunter at NWIC for expertly isolating and culturing this sample before I stole it. Terry King, whose work with yesotoxin is a big base on what we're doing. Clara Schliemann at Lummi Natural Resources, who gives us lots of clams to test for yesotoxin, and everybody at the Salish Sea Research Center who has helped me a lot on this work. And Noah, of course, for the funding. Any questions? Yeah. Thank you.
I assume you've seen Bill, Brian Bill's paper on Alexandria. I mean, same study, and you can use that as your foundation.
Exactly.
Yes.
Yes.
So Brian Bill
Yeah.
did a similar study around temperature and salinity actually, relative to Alexandria. So that kind of Bill Coughlin's temperature bar is
Exactly.
is, you know.
There's a question here about if I've seen the paper about from Brian Bill about Alexandrian with similar experimental design, and exactly right. This is especially with the temperature gradient bar. It's
On Anderson's lab.
exactly. It's a strong scaffolding to build an experiment on.
Yeah.
Yeah. There's a question in the chat. Is there any impact of dissolved YTX on shellfish? That's a great question. Maybe. It seems like particulate is the biggest concern with shellfish, but there is some anecdotal evidence that maybe dissolved is a problem with other toxins. And so it's reasonable to think that yesotoxin is similar. Anybody else? Okay, cool. Thank you very much.
Awesome talk. Thanks, Megan. Both of them, yes. Okay, next we have Kate Hubbard, who was here helping us yesterday with our Corenia cultures and is now going to give us a little talk about her research. Did you give me
I loaded on.
Oh, that's right. I was gonna get it at lunch. Okay, sorry. This will take a minute for me to download the talk, but hang tight. Hubbard, there it is. Okay. Download. I think I was planning to do this at lunch and then got distracted chit-chatting. Share screen. You're good to go. And you can use the arrows to advance.
Thank you so much. Okay, so I thought I would talk a little bit about some of the work that we do that goes a little bit beyond light microscopy. And I thought I would just go through a few different event response scenarios where we've had to use these additional tools. And part of that will also explain what I plan to do with some of the samples that you guys collected last year. Before I get started, I want to thank, so I'll be talking about work that's happened over kind of my whole career. So there's a lot of people to thank. I'm not going to thank all of them, but I will thank NOAA and NCOS Event Response, the Woods Hole and Pacific Northwest Centers for Ocean and Human Health that are funded by NSF and NIEHS. And then I have a big team in Florida and I just want to thank all of them. I know you guys are thanking me, but that thanks really goes out to them. So just want to say that too. And then I'll get started. Maybe. I'm just there. Okay, and now the arrows should work. Okay, so some species are, as you know, hard to definitively identify with light microscopy. Anthony gave a great overview of Pseudonychia yesterday. Some of those life history stages of dinoflagellates may be tricky to identify. And sometimes we have an emergent event in an area where you have a new species that comes in. And so you really want to have definitive confirmation, especially if you suspect it's a HAB and it might be producing a toxin. So I use a lot of different types of things to look at the DNA. And so when we think about what samples I might use, I like to get pretty creative here. And we can work with live water. We can pick out individual cells, or we can concentrate that water and we can get DNA from it. We also can work with Lugol's fixed material, and this is something we've been developing where we can pick out single cells or we can filter that and we complex out the iodine that makes Lugol's brown because that can inhibit some of the reactions further on downstream. One of my other things that I both love and hate to work on are feces. So a lot of times if wildlife are involved with an event, we may not know exactly what's going on. We may find toxin in these animals. And so we can actually test their fecal material to get a better sense of whether or not they may have been ingesting certain HABs. And I won't talk about that work much today, but we've worked with bowhead whale feces and also sea turtle feces, and we have been able to identify Pseudonychia in those samples. So some of the things we do for DNA extraction, we can do a full regular DNA extraction, which involves a kit and a lot of reagents. But sometimes when we're trying to get a quick answer, we can do something as simple as freeze and thaw cells a number of times. And then I can go from that into a series of different steps to amplify the DNA. And then from there, there's also a lot of different types of analyses we can use. Cheryl gave a great overview of qPCR, quantitative PCR, but we can do things like DNA sequencing. We can do things with antibodies. And so there's a lot of flexibility. And I like to think about molecular biology as being sort of a toolbox. And so if we have a new HAB issue, existing methods may not really be sufficient, and so we may have to adapt things to try to work with, getting exactly what we're trying to get. And that's kind of exciting sometimes, but it's also usually pretty tense because there's an event going on. So this was an event that happened in the Gulf of Maine in 2017. I mentioned this yesterday, but this was one of the Carnea mikimotoi blooms. And the first bloom that was really reported there was in 2017. And these are the samples. And so this is in Portland Harbor. And so just in terms of getting samples that may raise a red flag or a brown flag, these obviously did not look like their normal samples. And what we saw with my iPhone, I'm not nearly as skilled as some of the people in the audience here, was approximately 35 million cells per liter of the species, so tons. This species, in terms of impacts, there weren't widespread impacts reported, but it can cause issues in terms of dissolved oxygen consumption, and it can also sometimes impact benthic species. But fortunately, there wasn't a lot going on. And look at these cells. Do these look like the cells that we saw yesterday? They look cruddy. They're not happy. There's so many of them. And at that point, there must have been some sort of physical aggregation to reach those high cell concentrations. So what we work with, we have several different assays that are specific to Carnea brevis, just because that's such a prominent species in Florida. And so when there's a Carnea incident in other places, one of the first things we want to do is use those Carnea brevis specific tools. And so we were able to use qPCR. We did not get any amplification. And so that was helpful to at least rule out that species. Then I was able to do some DNA sequencing, and it's a little bit tricky to see this. I'm not going to try to move my head, but basically what we can do is go in, and I was able to get some DNA from the sample, and then I was able to sequence that DNA. And maybe let me take a quick step back, because we have assays for Carnea brevis, but we didn't currently have assays for Carnea mikimotoi at this time. And so we were able to look at sequences that are in public databases, design an assay to pull out only Carnea mikimotoi. And so that's what I did to generate these sequences. So I used a little bit of a sequence that's specific only to Carnea mikimotoi to help pull out this gene. And one of the things that we saw when we looked at this is that there are different populations of Carnea mikimotoi. And so when we looked at what we saw in the Gulf of Maine, it grouped with other species from UK and Norway. So that was pretty interesting, and that made sense in terms of it being in temperate waters. We see that our Gulf of Mexico Carnea mikimotoi groups with things that are sort of more similar in terms of latitude and water temperature. But again, Carnea mikimotoi is one of those species that is very broadly distributed, and it does form these very dense blooms. And so that's just kind of a little taste for what we will probably do with these samples, where we'll try to pick out some cells. We may also try to look at a bulk DNA sample through filtering the Lugol sample. And then we have some Carnea specific primers, and then we have species specific primers. So we'll try to rule some things out, and then it's kind of a learning process. So if we strike out with a few different species, we'll keep going. Okay, so that's a little bit about Carnea. I also wanted to talk about Pseudonychia. And so Pseudonychia, we've heard, very broadly distributed. There's about 50 species, and we really can't tell the species apart very easily. And as you know, some of these species produce DA and some of them don't. And we've had shellfish harvest closures in many different states at this point. And these have been tied to different species of Pseudonychia. And the first event was in 1987 in Eastern Canada, but it wasn't until recently that those dots appeared on the East Coast. And this just shows some of that variability across the different species. nutrients and salinity in terms of toxicity. And so identifying species is a really good way to think about their potential for producing toxin. But if you look in the green and the red, we know that nutrients, salinity, and lots of other things can cause Cyanotoxin to become toxic. So it's kind of a weird one. And some work recently in the Arctic showed that a species that we thought was non-toxic began to produce toxin in the presence of copepods. Those are Arctic species, and that relationship may not hold everywhere or for every species, but it was certainly interesting to have this non-toxic species suddenly become toxic. So unfortunately, like, that's something that's on the radar. So one of the things that I developed a long time ago was a DNA fingerprinting method for the Pseudonetia species. And basically I can amplify a region that is very length variable across the species, and I generate a fingerprint. And so each of these represents basically a different sized chunk of DNA that corresponds to a different Pseudonetia species. And so I've been using this method for a long time, and I know which species these are. And so I can go into a sample, do this fingerprinting, and get a good sense of which species are there, and also which may be dominant. And so I did a lot of this work in Puget Sound, and I just wanted to mention that while I was working on my master's degree in 2005, I was getting ready to defend in October, and getting ready, everything's going well, and then we have these 2 closures in Puget Sound. And so I was able to go out and sample those closures and present that data at my master's, which made it a much more exciting presentation, but was not great for Puget Sound. And what we found was that the Squim Bay bloom was dominated by Pseudonetia cuspidata, which is one of the small-celled species. In contrast, we had about a month later a bloom of Pseudonetia australis in Penn Cove. So it was really interesting to be able to apply the genetic methods and develop them further using these samples. And then I was able to go on some opportunistic cruises and use this method to look at a lot of other samples. And so I will show just a little bit of that from one of the PRISM surveys that I went on in 2008. Take 10 years off of that. And so this is just showing sort of a depiction of Puget Sound going from the Strait of Juan de Fuca through Admiralty Inlet into the main basin, and then into the South Sound through the Tacoma Narrows. And so this is showing potential density. So this is just driven by the temperature and the salinity. And one of the things, when you can fingerprint all of these species, you can start to see really interesting patterns that correspond with the hydrography. And so what I'm showing now here is the genetic data. So we have the different species that were observed in the two.
We can maybe hit it on the way back if they come back.
Trent has something to say. I think he's got, I think he sorted it out. And so what we're able to see also is the relative intensity of these.
They charge your card when you get there, and then they take it off.
I'm still talking.
Do they charge the tax, or is there's no tax?
Okay, we're good. Sorry, go back to here. Okay, you're good.
We won't put Trent too much on the spot for not watching the Pseudonetia stuff. Hopefully he doesn't get a bloom in his area. Just kidding, Trent. I don't know Trent at all, so. Okay, so we have all of these different species, we have the different proportions, and then as we move from the Strait of Juan de Fuca into Admiralty Inlet, we see that there's a big transition in terms of the species composition, where we have domination by Pseudonetia delicatissima, and that kind of changes into Pseudonetia pungens, which dominates, and then interestingly, we have both sort of co-dominant at the Tacoma Narrows, where there's a big sill. And then we see, again, as we move through South Sound, we see these transitions. We have a few other species here, Pseudonychia multiseries, which was present in a lot of the samples, but not at high levels. We also see, if we look at this assemblage that's out in the street, we don't see all of those species within the sound. And some of those species like Pseudonychia australis and seriata, which unfortunately I can't differentiate with this fingerprint, those were present and mostly in the Strait of Juan de Fuca, but we did see little hints of them throughout the sound. So that just kind of says that these species are everywhere. There is some structure due to the hydrography, and also probably due to the environmental conditions present in the sound. So this was really interesting, and to see these dynamic transitions just in this one snapshot, it gives you a really good sense of how important these sills are for circulation in Puget. And so they can act as kind of a barrier, and they can act as mixing zones. And I just kind of go through that a little bit more, where at these sills you can have upwelling, localized upwelling and downwelling, and that can lead to retention, and that may help contribute to those differences in terms of the species assemblages over longer periods of time as well. Okay, and then the last one, so in 2016, we have been wrapping up a multi-year field project in the Gulf of Maine, trying to look at Pseudonychia there. And it was an okay project. We had a little bit of toxicity, some different species changing seasonally, but then wham, we saw really, really high levels of toxin. There were shellfish recalls, and it turns out that Pseudonychia australis showed up for the first time ever. And you all know that's a really bad one on the West Coast. We still don't know the origin of this, although we've done some physical modeling to show that it must have come from a more northern area. But whether that means it came across from the UK, we're still not sure. This was one where we had never seen it before, so we were able to do a combination of scanning electron microscopy and then picking those cells. And our electron microscopist and I literally got to P. australis at the same time, and we were heading for each other's offices, and we were like, I don't believe what I got. And she's like, I don't believe what I got either. So we were like, okay. And we got the same thing. So there have been blooms of this organism since 2016. Things seem to have calmed down a little bit, but it's something that's still detected occasionally in the phytoplankton assemblage. And so we have, and we were able to verify this and compare against Pseudonychia seriata. They're a little bit different on the East Coast, so we can actually differentiate them. And so we were able to see that prior samples had had seriata, and that we saw this australis just kind of pop up. And this just kind of shows the Pseudonychia abundance in cells, along with particulate DA and DA in shellfish, just to show how quickly it jumped up. When the state of Maine looked back at the cell numbers and the toxin numbers after the event, they figured out that an action level was around 3,000 cells per liter in order for them to stop the event. So that's something that they've kind of kept going. 3,000 cells of Pseudonychia is all the time. So it's kind of a tricky one. And then, so we are continuing to expand this toolbox, and there's new rapid screening tools. We just heard about a lot of the different toxin screening tools and how those change over time. And so one of the things that we've been working hard on is trying to identify ways to specifically identify Pseudonychia australis. And so we've identified, you don't really have to look at this too much, but we've identified qPCR methods, and then we've adapted those for CRISPR. And one thing that, you know, COVID was terrible, but there were a lot of these rapid screening tools that suddenly became much more mainstream. And so one of the things that we're looking into is how this can be adapted for rapid screening. And so we have a big team looking at this as part of a ORHAB project right now, but they've been able to do PCR in a coffee cup, for example, that has a little heater on it. And we also are working on potentially converting the CRISPR assay into a lateral flow assay, similar to something like one of those rapid antibody tests. So fingers crossed that that will be effective. One of the things that's great about the CRISPR approach is that we use a lot of different probes in the development, and so it tends to be really specific. And I think that, yeah, that's all. Thank you.
We have some time for questions, if anyone has any for Kate. Mark in the chat shared something.
Yeah, sorry, Michelle, Mark. I don't know if you guys can hear me okay.
Yes, we can.
Cool. Yeah. I just, for people's awareness, Kate just mentioned a MARHAB project that she's involved with, led by Gloucester Marine Genomics Institute. And that's just a project link. You can share it with folks if they're interested in finding out more, or buttonhole Kate, which is even better.
Awesome. Thanks for sharing that link.
You got it.
We also have a message from Vera. Kate, did you positively identify K. mikimotoi in Washington samples? What risk does it pose to this region? Thanks and hi. Sorry to miss yesterday's. Okay, that's separate. That's just a personal note.
No, I have not yet worked on the Washington samples. So that's kind of the next step.
Because they're still in our fridge.
Full disclosure, they're still in the fridge. There are some storms and some other storms, and we don't like to ship cross country in bad weather because we've had things get stuck for 2 weeks. So we're really picky. And then, is anyone working on the pathway for toxin production? Yes. So recently the domoic acid biosynthesis genes have been identified. And so this presents a really great opportunity, just given how variable toxin production is, to get a better sense of when the cells are actually expressing these genes. And they do seem to be well correlated with domoic acid production. And so we've adapted this assay already for the domoic acid production gene, one of them. And that's working really well, so that we can look not just at the presence of P. australis, but hopefully what it's doing, and get a better sense of when it is actually, ramping up to produce DA. One of the things Anthony mentioned, P. australis can kind of front load domoic acid, and it can produce it throughout its growth cycle. Some Pseudonatia species will produce more toxin after they slow their growth. And so there's quite a bit of variability. And again, trying to understand that is an awesome mystery to unravel. And these new tools will hopefully help. There's also people doing transcriptomic and metabolomic analysis to try to get a better handle, again, on those processes and some of the drivers. Thank you, guys.
And I wanted to give another special thanks to Kate for coming out here, and also a special birthday shout out because she's spending her birthday with us. So happy birthday, Kate. Okay, so, well, that's most of our talks for today. And to round out the day, we're gonna have a couple of SoundToxins program updates, and then we're gonna end with a community discussion. So let me pull up those slides. I think they're here somewhere. Sorry, I need to find where I put them. I was very organized this morning, and then after lunch, I didn't get a chance to pull up everything. Okay, here we are. P2 program updates. Okay. Okay. Share slide. Video on. Okay. Okay, so program updates for this year. One of the main ones that we talked a little bit about yesterday, but for everyone to know, we're making azidynium and crenia, which we've talked about before in our program and said, you know, look out for these, but we're boosting their look out for me status to required reporting. So this update hasn't hit the database yet, but probably in the next couple of weeks or so, you'll start seeing them in addition to the 7 that we currently have as like kind of mandatory reporting. We were really lucky to have some cultures yesterday of both of these to look at to kind of try to get better at identifying them. Azidynium is particularly difficult because it's so tiny, but we have some footage of that on the recording. So if you missed it yesterday in person, we'll send out the recordings. They might take a while since they're going to be such long videos of like the entire day to process, but we'll try to get those recordings up online so people can go back and watch them and practice and study that. So, and crenia as well. So look out for those. We're also doing updates to the database to include a lot of like new species that haven't been there. And also, as I mentioned yesterday, we're going to be updating some of the species names to be more up to date with the times. So I know this can be a little bit emotional for folks. So I did, oh no, I hope my media works. Hang on one sec. I have to enable the content. I forgot that I had to do that. Okay, so I made a little tribute. See if it works. Wait, hang on, I have to share the slide. Sorry, pause for the effect. Everyone in the room's giggling because they know it's coming. Okay. All right, let's see if this works.
Will you remember me? Don't let your love pass you by.
Okay, so these are a few of the main ones that I found particularly difficult. But we will be releasing a full list of new species with their names. And there's a few that the whole genus has changed. So like Serratium is tripos now, all of them are. But then for a few of them, like Dictyocha speculum is now Octactus. Oh, I think I might have spelled that wrong. Octactus, there's another C in there. Sorry, if you're taking pictures, I'll edit this. Octactus speculum, but there are still some Dictyocha genus or species in that genus. So a few of those are different. I think Rhizosolenia also, there are some that are still Rhizosolenia. It's just Stetigera that is a new species. So again, it's not 100% ready yet, but I will be sending out some resources at some point in the next couple of weeks. That's like a full updated species list. And when we go to update them on the database, what I'm thinking of doing, and we'll see how like clunky this is for like data analysis and input, but I'm thinking of having tripos parentheses like formerly Serratium just for like a little bit. So as you guys are inputting data, you don't have to like memorize all of these right away. We'll give a little transition period for those. So that's the plan for those, and we'll share that in the future. So chat. Okay, so those are the big updates. Oh, yes, question?
Do you just have the Dictyocha speculum called in this painting?
Mhm.
Is it also Dictyocha fibulum?
Nope, that one's still Dictyocha. Yeah. And yeah, same for, what's the other common Rhizosolenia that we see? Does anyone know off the top of their head? That one's still Rhizosolenia. Styloformis is still Rhizostonia, it's just this one that's different, which is, yeah, especially annoying. The other one I didn't include on here is Odontella mobiliensis, which we saw blooming recently. Kind of, it was a big deal in 2023. I was like, oh, it's the first time we've seen it blooming. That one is Tires, but all the other Odontellas are still Odontellas. So yeah, this is not comprehensive. This was just the ones I threw together with my little movie last night. Thought it was funny. Okay, so those are the major updates that are like definitely gonna happen in the next couple of weeks. And then for the rest of the time that we have, we wanted to move into like a bit of a community discussion about some potential changes that we could make, but we want feedback on. So I think that's the next slide. Melissa, do you want to chat a bit, or
We can go either way.
Okay.
We kind of just wanted to have a like full discussion with the room of like, I'm really new. I know Michelle's only been here for like 3 years, but some people have been here for much, much longer. So we kind of thought of all of you guys as the specialists that can help us make the changes to the program. So this one was just wanting to go over the original goals and if we saw any changes in those goals. Or if people had any thoughts of like, oh, wow, we don't really do that, or maybe we do. So the main goals that are listed, this is listed on our website, which is a little out of date. We're working on that too. But to determine which environmental conditions promote the onset and flourishing of HAB events or unusual bloom events. So definitely, I think we do that. To determine which combination of environmental factors can be used for early warning of these events and to document unusual bloom events and new species entering the Salish Sea. Do we feel like we do more than what's listed there? And do we want to list things that we maybe work on? Any thoughts?
And just to add to this, one thing that's cool is that I think the program originally started in 2006. So next year is a 20-year anniversary. That feels like a good time to kind of like reassess the program and just like make sure things are still going well. And also if some of these questions are catching you by surprise and you want to like think about them for a little bit, we're planning on having like a feedback form that will have some like opportunities to answer some of these questions after you've like thought about it. So we don't want to put people on the spot here, but just kind of thought questions. And we have a few other slides to think about. And also to add to this, I think there has been advisory committees in the past that have helped inform like SoundToxins, but there isn't currently one. So we're hoping to create one as well that's going to help put like scientific inputs into the program. But again, before we make any like big broad changes to the program, we want to talk to the people that are actually doing the work and make sure you guys are included in the conversation. So that's probably kind of part of what this is. So
if we don't have any changes to the goals, that's great. But if anyone has any thoughts, yeah, that's it.
Well, I'm just gonna tell you from our perspective as running a nonprofit organization, we got, we are managing that, supporting volunteers and providing space. That we greatly appreciate having a common database and data processing so that we can contribute to the science. I think that helps a lot, and I think it's also great for, as a means of our volunteers to be able to get engaged and learn and share what they know and all that. We entered under funding, and that got us up and running and going, and then the funding disappeared, and now we're just like running on fumes. And I know everyone's having a hard time, but I just have to say that that's important because we need to keep paying our electric bills, stuff like that. So we love being involved. We're just do our best. We appreciate getting equipment when we can. We appreciate getting all that stuff very much, but it does help.
Thank you. Definitely noted. And I think the kind of permanent funding of SoundToxins is something that's been on our minds as well. We're currently grant research funded only, and those have an end date to them. And so we definitely want to make sure we can keep us funded. Also, big shout out to Nanus, who gives us a little bit of money to keep going. It's not fully funding the whole program, but that's definitely a good source of money, but something to kind of definitely think about and talk about. So thanks for bringing it up, Betsy.
Tracy's conversation yesterday, a couple of other people brought it too. Notification, when you see a bloom or whatever the yard is. So the surveyor sent it up to whatever organization, gets up to you, it's free, then you have to make, instead of a million calls. Why create something a lot more economically viable, you build, if you think on your cell phone or about, your cell phone diameter words, you get things, all kinds of things, so you only have to make one call, and you have to maybe subscribe to it, because probably 10% of Washington, West of Washington would have something like that. That would cut your expenses down, cut your time down, and everybody gets the same message at the same time.
Yes, great thought. We kind of tried to do that through loss of hearings at some point, like just like you get the tornado warnings and there's the Olympia occasionally. We couldn't quite figure out how to make it work at this time, but yes, great thought, beautiful thing, even an automated email system or text. Yeah, haven't quite sorted it out yet, but yes, great thought, future, beautiful piece.
On the wish list of items. Great idea.
Just before we move on to this next slide, I just wanted to add to the second bullet of kind of the prediction and the early warning events. One thing that I was gonna bring up earlier, but we were late for lunch. Cheryl, another one of the many projects that Cheryl's working on is a project to use the Salish Sea model to predict HABs. Do you want to talk a little bit about that, Cheryl? Is there anything you can share with us right now about the status of that project?
Yeah. It's just starting now. And he's got the Salish Sea model, which is an FEPON model. And what they're doing for this particular one is looking at, I think they're building a water quality through the coliform. And also we're going to build some sort of map prediction. It's pretty limited, it's the first step. And they're not gonna have results for like years, but that literally is just for about the funding in fall.
Yeah.
Until they start March. And then, you know, you could have one day it's gonna be, you know, find out one or 2 minutes of the HABs, they already have some of that model. You know, we'll have more in about a year's time, but right now it's just a very beginning. So.
And just to repeat that for the folks online, in summary, there is a project that Terang's group is working on out of PNNL, with, as well as UW Tacoma. But it's at the very beginning. They just got funding in fall and they're starting kind of now, but is early on, and maybe we'll have more information about that going forward. Oh, great question, Rick. That will queue up maybe our next, actually, I don't know what order these are in. I'll go ahead and read the question. We're talking about the data policy about proprietary data. Should our data be available for research as it's related to our goals? And we definitely were going to talk about that.
It's a few slides down.
It's a few slides down, so we can jump ahead and talk about that now if we want. Yeah, okay. Okay, so I think we started talking about this a bit last year, but didn't really make a ton of progress, but now is a good time to restart that. So the way the program was established was that we are a partnership with a lot of different groups with a lot of different priorities and needs. And so when the work was started with the partners that were there earlier on, it was decided that the data would be privately collected for partner use only. And then since then, if other folks want to use the data for research, it has been a like case-by-case situation where people ask for data from a certain site for a certain purpose, and we get permission from the site's data collectors to use that data and then have provided data for that. The question about whether, I guess, so we've also heard that a lot of people do want their data to be used more broadly, and that's kind of important to us. So earlier today, Cheryl was talking about the phytoplankton vital signs. Using that, contributing to that database means that that data would then become publicly available. So that's one good way that we could get for the folks that do want their data to be used widely to engage in that process. It's helping a good mission and research. And then also has added benefit of then data being available more publicly for other people to use for research and analysis and get that data out there and more widely used. So that's really great. And it is going to be, I think what we're going to probably do is our database is probably going to stay private. And but if anyone wants to contribute to the phytoplankton vital sign, that's a really great way to get that data like publicly available without having to manipulate our database to do both, if that makes sense. So I feel like that's a pretty good solution that also protects the folks that don't want their data to be public. And again, if you're part of the partnership, you can still, see and access that data to help inform, your decisions, what the sites around you have going on. But then for those that do want it to be public, it'll be publicly available. But I'm open to like comments and thoughts about that, because I know we were thinking and talking about it a bit last year, and I was trying to figure out like what the best option would be to have some of it publicly available and some of it not. And then Cheryl was like, hey, well, we're going to host it on this other platform. And I was like, that seems like a good solution, and I don't have to do extra work except for hand it off. But yeah, definitely open to hearing comments from folks about how they feel about that. Happy? Good? Satisfied? Okay, I'm getting some thumbs up, so that's great. We don't want to put anyone on the spot, but there'll be an anonymous feedback form that you can share your thoughts more openly afterwards if you want. Anyone online want to chime in? Rick, did you want to comment on that, or does that sound good to you, since you were the one who asked the question? In favor of more transparency. Yeah. And do you think that, or hopefully this working with the PSEMP vital signs is like working towards that goal? Yeah.
We also have a little bit more staff capacity, so maybe we can write more reports that are publicly available, that are going out to all community members later on, not in the moment, maybe a year, like a year report or something, just so that then more people could be accessing the data and see what's going on.
So our next kind of big question is about our protocols. So they, again, were set up to be really rapid. Like I think, so I've heard that like early on, they were taking like 1 or 2 people were taking samples just like once a month at all these different sites. And they were like, this isn't enough. It's not enough granularity of data for just sampling once a month, and also not one person can do all these things. So let's turn this into a program modeled after ORHAB to get communities to help collect that data and have it be more frequent. And so to do that, they came up with a protocol that was going to be easy to do and use, even if you don't have a full lab set up, and with minimal equipment and training, and quick, so that if you are doing this on top of your other full-time job once a week, you have the time to do that. So that's where like relative abundance versus counting every single cell and doing like a complete community composition comes from. And that's also where kind of the idea of the net tow and the whole water sample and the way we do that, even though it's like a semi-quantitative count, that was kind of thought to be the like kind of fastest, easiest way. But we also know that there are a lot of problems with that. If you have a really strong current, if you don't have a flow meter on the top of your net, and even maybe if you do, there's a lot of like flushing that could happen. There's a lot of extra water that could be going in if you have a strong current. The rate at which you pull the net can make a difference. And so there is definitely some, and also like rinsing the side of your net if your net gets clogged up, and stuff gets stuck to the net rather than getting into the cot end. Like there's definitely several places along that process that variability can come in. So, is that method working for us? Is it effective at giving us the information we need to do the biotoxin predictions? Is there a way we can make it more scientifically robust without adding like a ton of extra time and work? And then a big one that has, well, one thing that's come up frequently is just the like variability even at the qualitative scale of relative abundance. And some people want a little bit more guidance on like how to group things into present common bloom, especially when you're comparing different cell sizes and then like winter to compared to summer samples and like how dense things are. So kind of trying to make that a little bit more specific. And then another big one is this sample preservation. So formalin is toxic and kind of an icky thing to use, and a lot of people do not want to use that. There are a lot of good uses for it, but also is that necessary for us to be continually buying vials and giving them to you and you filling them, and what are we doing with them afterwards? What are we using the long-term samples for? This is a question that we want to talk about because we don't want to waste anyone's time, but sometimes it's really great to have historical records. So yeah, there's a lot of things to talk about there.
Yeah, that was a lot of questions. A lot of questions. If anyone has any thoughts on any of it, we'd love to hear. Yeah.
Big question. Well, not a ton right at the moment. Repeat the question, thank you. The question is, what are we actually using the preserved samples for? And the answer is that like we're not doing a lot with it right now, but there are some things that we could do with it, and there are some reasons why it could be a good thing to have. So some of the reasons are, let's see.
Yeah, well, we also, for QC, I think was the initial reason, like we could look at the samples and then actually kind of have an idea of if the counts were similar to what we were seeing preserved. So that could be a QC protocol. Right now, we're not doing that. Hypothetically, we could bring it back with more staff time on board.
Yeah.
For every sample, absolutely not, but like, you know, every once in a while.
Like spot checks. That's one of the ideas. The other one was like,
eDNA?
Yes, yeah, that's the next one. So if there was like a specific event or something, then we could go back and like target like, okay, let's double check, you know, leading up to this, can we see like the rise in cell counts or something? That's another reason to like keep them just in case. And then the other idea that we have had and thought about is we can use those preserved samples for more robust like eDNA analysis. So there is a way to use formalin preserved samples for eDNA. And then for a bit, I was like, oh, wow, this is really exciting. We have all these preserved samples for like going back quite a bit. It could be really cool to get some analysis for this. And I thought, but who's going to do that? Where's the money going to come from? But we actually are maybe starting conversations with some folks at UW about that. So I will have more information later this week or soon about the feasibility of that. So that's one thing that could be possible. The other questions that I have around that is like, okay, do we pick a couple of target sites to do that? Do we do it for all the sites? Is that still necessary? And some other pros and cons of formalin is that historically, that's one that's pretty good at preserving most of the things that we see. It's pretty good at preserving diatoms and certain thecate armored dinoflagellates, but it's less good at preserving a lot of other things, including this, you know, new threat of Karenia or other unarmored dinoflagellates and flagellates that just can pop when you see them. The idea of using lugol's has also been an idea. However, that is good for helping identify some things, but also has its drawbacks at not being super long-term. Like eventually the lugol's fades and you have to refill it, and that becomes like a full-time job in itself, especially at the scale of what we're doing. So yeah, that's some information.
Yeah, does that answer the question of like, right now, we're not doing a ton with them, but there's a lot of potential. And I think we have some nervousness of like canceling it or not, you know, not doing it anymore and then being like, oh, wait, we want to do eDNA. And then we have a big game, like we're not doing it for years anymore or something. So I think that's our concern of stopping.
Yeah. And also I needed to find somewhere to store all the samples recently. And I was like, I'm just going to throw them all away. And then I thought, oh no, all this history, like maybe there's something we can do with it. So we did find somewhere to store them in a basement on UW's campus in the old oceanography building. But so we do still have those, and hopefully we'll find something to do with them, because it would be great that they would be, you know, useful for something but yeah, it's not like we're constantly using them all the time, so we don't want to waste time or money either. Kate, I'd love to hear.
And we have been trying to find like target sites and like we just learned the sites for this to subset. And then we also have rules. So if there's this much of this thing, then we would see about it. So we have a rules that we follow. It's kind of annoying, but like, we change the rules when we find it helps. We have a small room. So,
Have you ever had oh, sorry, in case the folks online didn't hear, Kate shared that there are similar issues with samples storage in other areas, not just us, and that they have come up with a set of rules to decide which ones to keep and throw. There's like a couple of sites that are like kind of target sites that they keep, and then also if there's like certain cells or like at a certain abundance, then they are also saved. So it's great to hear there's like some like other models that we could go after, and maybe we just like kind of go on a come up with some rules for ourselves. My question for follow-up question for you, Kate, is there ever a time where you've like gone back and really used those like older ones, and why and how might you imagine that we would do that at our program?
But definitely we need to go back and see, like, sometimes species change names, and sometimes you see something and it's not the same as this other thing, so we go back and look there and
Okay, that's wonderful. Yes.
Sometimes there's new issues that emerge too, like I know recently Christopher Clancy's been talking in the last 5 minutes about how the uptake in noctilues can decrease the species composition of your samples. And we started collecting noctilues cell counts in the last few years, but I've had like a student come in and want to look at our long-term data, and I've had to go back to like old samples to do the noctilues cell counts, and it was nice to have those.
Oh, that's a good one. Yeah. Yeah. So yeah, that's the kind of I think these are the examples of why we do it. And I just realized something while you were speaking, which was that for one of my concerns about updating all these species names was I was like, okay, great, we reclassify Rhizosolenia Setigera as Sundestromia. But now every time in the past that someone wrote Rhizosolenia SP, how do we know? But if we have formalin preserved samples, in theory, we could go back and like check. But again, that's a lot of man hours. So like how, like, and then like, okay, we would only do that if it was like a target species or species of concern, or that we were like thinking or questioning about. So there's still, I think, like some amount of rules we could put on like how many we keep and what. But then the question of like forward thinking, like, do we still continue giving everyone vials and asking you all to collect formalin samples and use formalin and keep it at your sampling sites, some of which are nice labs and some of which are not. Some of them are just your houses. That's kind of an open question. And we've had also a few people that have been like, I do not want to use formalin. And I've just been like, that's fine. You don't have to do that. Yeah, we have some in the room. And but then like, okay, so we're changing the rules for some people. So do we just do it for everyone? Like kind of when, at what point do we make that decision is something that we're like just struggling with deciding.
I would like the idea of having like certain standards of when you should keep samples. It just feels wasteful, like especially in the winter when there's like 2 like.
Yeah.
I think like having some sort of threshold would be helpful.
Yeah, and that would also save money because the vials are not cheap. And just to repeat that, it was that having some rules would be nice so that we don't have to keep samples, like especially in the winter when there's only like 2 things in the sample. It's not wasting a whole thing. So, yeah. Okay, love that. Oh.
Another way to keep records would be that people are cameras and just record the scan of the microscope as you do the tank and have that sort of thing. That really useful.
Yeah. Yes, yeah, using my cameras to capture things can be really helpful both for training and for record storage. That doesn't take up physical space, that does take up a lot of potentially hard drive or cloud space, but is an idea. And then just also, I know not every site has a camera that's good, and that's also something that I've been, sometimes I'm like, send me a picture if you don't know what it is. And sometimes it's a great cell phone picture, and sometimes it's not. So, so that's also something that I've been thinking about.
Climate change models are changing all the time as far as how long, because it's moving faster. Maybe you can lock in your sample, but how far back do you need to go back? You gotta go back to the dinosaur. So if you only care about the last 5 years, 10 years, where your current climate change model is, and say lock into that schedule, that would probably get rid of an awful lot of stuff that's 25 years old.
Yeah, we don't have stuff that's that old. I think I just moved the sample, so I could tell you like the oldest ones. There was like a couple from maybe 2014, and then mostly a lot more from 2018-ish time frame, and it's not for every site. So, but that's a good point, that like looking at the time that we, yeah, how old it's old, and what do we care about.
Any other things about our protocols?
Speaking of photos, as far as like relative abundance goes, and we have that little chart with the ABCs defined. And if there would be a way to maybe use photographs from, you know, the last, like from samples that are really good at showing that difference between fun and blue and all that. That might be helpful for training new folks so that they can actually see it on the slide what it looks like.
Yes, absolutely. The question, or the question comment was that it would be helpful to have better training tools for abundance rather than the ABC picture that we've used forever. That's not great, but to actually use microscope pictures. So two comments on that. That definitely, I agree that like if folks can send pictures of good community compositions, we can try and do that. There are some, I talked to Anthony about this actually specifically, and one of the hard things is like, and people brought it up in the training, like, okay, if you're just looking at one viewpoint versus like the whole slide, kind of might affect things, but also I still think it can be a good, like, imagine this is your whole microscope slide, like, what does that look like? And so, yeah, I think we can come up with some better training options for that, and come up with some like kind of percentage guidelines to help people with that. I think probably the limiting factor for me is that I'm not on the microscope every single week, which I probably should be to keep up my skills, but just don't have time. So if you all who are actually doing this every week have like good pictures you can send, that would be really helpful for me. Paulette first, and then Amy.
I think relative abundance standardizing, even, but well, we certainly do it within our group because we have a lot of people. Nobody does it the same. When I've talked to other groups, nobody's doing it the same way. And we would all be doing basically the same thing. And the hardest thing for us is, okay, Fresno, we can say it's just a few, and Bloom, if we see it in every field of view, but common, that's a huge area. I mean, what does it mean?
Yeah, yes, yeah.
Can we give those percentages, or is that not? That's realistic.
I
don't know how they put it,
Anthony. I feel like Anthony, maybe you want to comment, because you were the one who gave me some comments about this last time I asked you.
What you said is true. So as far as being accurate, yeah, common, because of that is a big area. Yeah, room is dense, you know, it's a major component of the assemblage. Common is fine. Relative abundance, so yeah. Relative to what you're looking at on the site.
Yeah, I just think that's, you know.
And is there a difference in how you assess like diatoms versus dermatologists?
No, not really. You can compare them kind of against each other. But it's like a Bloom of Alexandria, it's not going to be like a lot, right? It's only going to be themselves. But if you do that, you don't want to put a note somewhere like, you know, Alexandria was blooming, but I saw it myself.
That's not 25 cents. You can do that. There's the next sample is because there have species where you have really miss things low, but also you can't compare to anything else that's in the sample.
So again, I know that this has come up several years in a row. I think relative abundance is something that we should improve the training on and try to come up with better like guidelines and training tools for people. So that's definitely noted. And we will work on that. Relative, yeah. Did you have something to say? Did we miss you?
Oh, yeah, and I'm talking to Joel about this, but I wanted to bring up the whole group. We do quite inlet, and it's about pseudo-niche accounts, the net toes versus the whole water samples. And I feel like we do our net toe, we come back, we immediately do our self-counts, we enter it in right away, unless that data's in there. And then in the comment section, I always say like, whole water sample preserved and settling. Well, they sit for a while settling, it's not 24 hours, sometimes weeks going down the jars. And we like the big hustle of like, taking the time to like suck off all the sample and everything. And then those vials will sit until I'm like, oh my gosh, we have to give our samples just a whole year's worth of. Have the dishes river coming right in, so when we're going out the water sample, it's like fresh water almost, and so I just get zero after zero after zero, and it takes so much time to do all those samples. And I have like 3 years worth of data where I have to compare next to each other, like the net toe samples versus the whole water. I don't know if it's worth it. It's a huge amount of time. And then afterwards, now I have to data, go back into the SoundToxins, and I'm not sure how I replace it. Do I replace all the drop down net toes and erase it and put in, now it's my 10x full water, I enter these comments. But what do you do? Because I know I'm not the only one doing this. Do you all go back and then like, do you put in your data? Or, I don't know. I don't know if it's worth it. Doesn't feel like it's worth it for me, but I haven't really compared the data. So.
Yes. Yeah. And so other people recently have also asked like, why is it that we do like pseudo-niche from the net toe? And Terry had said that there has like been like historically a better correlation with like domoic acid and the whole water sample rather than the net toe compared to everything else. And the net toe is good at aggregating things. But then when you say that you're seeing zero. Yeah.
Maybe it gets done just when you have a bloom. I don't know.
Yeah, maybe. That's something to think about. Do you have a comment, Tom?
I'm really probably able to out your different data for us, but we also take accounts management in our net. But we don't necessarily, when we put it in the data, we don't just drop menu down for pseudo-niche as net. We simply put what our net cell count was in the comments. And then when we do our whole water is when we go back and do the whole water difference. We're not having to replace, we're not entering the data and have to replace it. Like that cell count goes down below. And that's what the beautiful like pseudo-niche count and more. So do that part, in our net. When we do get tons of our net, we only come for half, and then we're kind of just hold on to you, and we're like, in our net.
But that's just a comment, and we note that we did that, so it's like, yeah, it's a rough. As far as actually, dealing with the jars, we just do it weekly, and granted, I have to let 3 days staff, so I do it 3 days later, but, perhaps it might be a thing that is, could do that before, and so it's a procrastination, is our problem. Yeah, it's rough.
Once a month thing, or.
Yeah.
It's a task.
Okay.
I'd love to hear what you're kind of like talking between your net and your whole water over the years.
So what you say is the whole water samples collected from the surface. I don't.
It's a good conversation. Oh, I already did that one. Already did that one. Okay, yeah. So great conversation on this. I'm trying to think of like the best way to like continue this. I think one thing that I have mentioned we're going to do is like an advisory council, because I think this is not a decision that like we're just going to make alone and then like enforce on everyone. I want like input, but both from some scientists to tell us like, is this worth doing the extra thing or not? Is it worth making all of your monitors do this or not? But also from you all to say, okay, well, this is really not worth it to us. Well, then let's try and adjust and change. So So, okay, thanks for all those comments. Again, if you have any additional ones, we'll get a feedback form. But this has been really helpful for us. Thank you. We already talked about data sharing. The next kind of feedback that we want is like, is there any program support that you should be providing that you're not receiving that you would like? I'd love some feedback on, we've had like office hours in the past, but trying to dial in like what a good frequency of that would be, just so that there's not just like empty Zoom rooms, but also that we're available when you need us. Site visits, like some sites I've been to a couple of times, some sites I've never been to. We'd love to kind of equalize that and see like how often should we be visiting your sites, and that kind of support. The data reviews, what do you guys want to see? Like what kind of analysis should we be spending our time on? How frequently do you want that? Is like a monthly good? And like, is that actually useful to you to get that data report? And what else would you like to see better? So those are all things to think about. We are kind of late on time, so maybe you can, if you have some strong comments, I'd love to hear them. I'll be here for like a bit longer as we clean up and stuff, but just to make sure we're not hanging on to too many people for too long, you can also give this feedback through a form. And then the idea of like a community forum for ID and other things to just chat amongst ourselves. We had thought about a Slack a while back. Slack is what we use at our work, but I know that not all of you might be familiar with that or want to download a whole new app. Trying to think of like another way that we could connect. I know some of the groups have already connected, which is so great. I love to hear that like Port Townsend and Camino Island and the Whatcom County folks have all like kind of connected on their own. If there's a way that we can kind of foster that connection between all of us, except for like aside from these community once a year meetings, that would be cool. That would be fun. More connections, it would be great. So thoughts on that, we welcome. And then, oh yeah, I already mentioned that we were gonna make an advisory committee. If you have thoughts about it, if you want to know if you have recommendations on who should be involved,
Or if you're like, man, I really want to be in the advisory committee, come tell us right now and we'll add you to it. Yeah, I guess we can, I know it's like the end, so I guess we can try to maybe do like an office hours or something too.
Yeah.
And just get some of those on the books so we can have these conversations just a little bit more regularly and people can pop in if they're free. And if not, that's totally okay. You can message us, call us if you have ideas.
And then feedback on the training this year and what you want in future years as well, just so that we can kind of dial that in. But yeah, so look for an email. And then if you're not on the listserv, like definitely let me know and we can get you added to make sure we're getting to everyone. But yeah, thank you very much. Unless anyone has any major comments, this is the end of our annual meeting this year. Thank you.